Dr. Dipendra Kumar, could you listen to me?
Yes, I can hear you. Good morning.
Great. Thank you. We will soon start. So could you make sure your presentation view and screen share is working with us? We'll start session in 4 minutes. Maybe it's better. Participants online and in person for the second day of the 12th ISAJ Symposium at the Kota University. This is our session 2, Room B, and this is a very special session for the life science students. And we have speakers, first speaker, Dr. Terry Kumar from the University of Scuba and of his topic sleep. And I am the session chair, Santosh Gotwal. I welcome you all and request invited speaker to keep talk in time is 12 minutes and 3 minutes for question and answer, total 15 minutes. So please, Dr. Dipendra Kumar, could you share your screen and start presentation? Thank you.
Can you hear me?
Yes, please start.
Okay. Good morning, everyone. Title of my presentation is Why is REM sleep important? In fact, sleep do not need an introduction. We all sleep every day. But let me introduce you briefly. Sleep is actually a universal biological need. In fact, we spend our 1/3 of our life in a sleep state. But where and in what form the need of sleep is encoded is not fully understood. As we experiences a day of sleep deprivation because of some reason, and we also notice the next day there is a more sleep, that is because of the homeostatic sleep rebound. But how is that sleep rebound is translated is not very clear. There is mainly two type of sleep. One is called slow wave sleep. It is because during that stage of sleep, our brain wave called electroencephalogram, so slow activity. Here you can see in the blue color. And the other stage is called rapid eye movement sleep. It is because of the eye movement during that stage of the sleep. During rapid eye movement sleep, there is a mixed brain wave, which is similar to awake stage. That is why REM sleep is also called paradoxical sleep. It's a paradox stage because body is in sleep stage, although the brain is like awake stage. In fact, REM sleep was discovered by Scalzi and Kleitman as a dreaming state. In fact, we dream most of our sleeping period during the REM sleep. But REM sleep duration is not uniform throughout a mammalian brain. Left-hand panel, you can see the animal which has a high REM sleep, more than 3 hours sleep, REM sleep per day. And in fact, platypus is the one which is the which sleep maximum into the REM stage. And the right-hand panel, you can see the animal which shows low REM amount, and human is somewhere in between these two category, which is around they sleep 2 hours out of total 8 hours of sleep. Even in human, the sleep amount, REM sleep amount is not same throughout the life. We sleep more REM sleep when we are young, and you can see when it's around 50% time when we are young, we sleep into the REM sleep, which reduces to 3.8% when we become old. So where is the need of REM sleep is encoded? We have started to understand the brain area which participate in the REM sleep regulation. For example, pedunculopontine tegmentum area, BL-PAG, or locus coeruleus. But in fact, we do not know where actually the need of REM sleep is encoded in our brain. So I will explain the possible function of REM sleep, 3 possible function of REM sleep. And the one is maybe REM sleep has some thermoregulatory function. This function we came to understand by an experiment in rat. So in this experiment, rat were given an option to select their preferable ambient temperature using this thermal preference chamber. The mouse can go to the one chamber to other chamber by connecting door. And what we found is when they were given an option to select between 3 ambient temperature, for example, 27, 30, and 33 degrees Celsius, they mostly spend time in the 27 degrees Celsius. But this was not the temperature where they sleep more REM sleep. It was mildly warm ambient temperature around 30 degrees Celsius. Rats slept maximum REM amount. This indicates that REM sleep also is sensitive to ambient temperature. Now, the capsaicin, which is a chemical in chili pepper, don't need a detailed introduction because we all know the Nobel Prize in the year 2021 was given to the one of the scientists for the capsaicin receptor discovery. Actually, capsaicin, when we eat capsaicin or capsicum, it stimulates warm sensitive neuron. In experimental situation, when the rat received a high dose of capsaicin, it can destroy the peripheral and central warm receptor. And what we did is we injected rats with high dose of capsaicin and measured their body temperature when they were exposed to various ambient temperature. So the red color is showing the mice which was treated with capsaicin, and you can see these mice completely lack the control of body temperature regulation when they were exposed to higher ambient temperature. The purpose of this experiment was to understand how much REM sleep will affected if we affect the body temperature of the animal. So here is the experiment where we selectively destroyed the capsaicin receptor at various level, like preoptic area, which is a sleep regulatory area in the brain, or peripheral and central, all capsaicin receptor were destroyed, or only peripheral capsaicin receptor were destroyed. What we realize is that destruction of capsaicin receptor or warm sensitive neuron attenuate the ambient temperature mediated increase in REM sleep. So you can see that the REM sleep was not increased in this mice when the thermoreceptor were destroyed. So by this experiment, we understood that the thermoreceptor has a role in REM sleep modulation. Let me move into the another function of REM sleep, which is recovery from the stress. In this experiment, you can see the x-axis is showing the time of immobilization stress in the animal. For example, 30 minutes or 60 minutes or 120 minutes. The y-axis is showing the percentage sleep rebound. The dark color is REM rebound. You can see that with increasing the restraint stress, the REM amount is increasing. This suggests that probably REM sleep has a role in recovery from the stress. I had an opportunity to study REM sleep recovery phenomena in a mice model of anxiety disorder, which is a Kaknak knockout mice. Kaknak is a gene which is expressed throughout the brain, mostly into the thalamus, and it encodes L-type calcium channel. It was from Jiva's study. It was reported that Kaknak is associated with various psychiatric disorders like depression or schizophrenia and others. As you can see in the wild-type mice, the restraint stress for 60 minutes induces REM rebound in the dark period, which was uh, much higher compared to the wild-type stage, but this rebound was attenuated in Kaknak het mice. So what we understood is if this mice is exposed to the chronic stress situation, they will develop the symptoms of depression or anxiety because there is no REM rebound in this mice. Let me quickly move to the another possible function of REM sleep. We all know that if we can't sleep, our memory will be affected. In fact, REM sleep has a bidirectional role in memory consolidation. Bidirectional means it can enhance or degrade the memory depending upon how much REM sleep you sleep after the learning. So in this experiment, in the first group, the mouse was REM deprived so that they can sleep more after learning. And in such situation, you can see the memory was enhanced. On the other hand, if the REM was deprived after the learning, the memory was degraded. So it suggests that REM sleep has a bidirectional influence on the memory consolidation process. We started to believe that memory actually reactivate during REM sleep. So upper panel is showing the home case situation, and the bottom panel is showing the animal which were exposed to the new context or kind of context learning. And when after learning or home case situation, they enter into the various stage of sleep, particularly the hippocampus, the brain area, which is important for memory, is showing reactivation more than during the non-REM sleep. This suggests that probably the cells which was activated during the learning reactivate during the REM sleep. But hippocampus is a big area in where precisely these memory related cells are reactivated. So we investigated a particular neural circuit called adult born neuron. As we all know, even our brain, when we are adult, they can create new neuron. Those are called adult born neuron. In fact, those neurons, as they mature, they encode memory, especially at the age of 4 weeks, they are more suitable to encode new memory. And later that, they cannot encode memory. So we targeted those adult born neuron during the REM sleep. We, using transgenic mice line, we express a calcium sensor called GCAM3 on the adult born neuron, and using a miniature microscope, we monitor the activity of those neurons during the learning and during the REM sleep and during the test. But we found that indeed the cells which was activated during the learning is reactivated during the REM sleep. So in the next stage, we wanted to know that whether those cells reactivation is really necessary for memory consolidation. For this purpose, using optogenetics technique, we selectively silence the adult one neuron activity during REM sleep. And we divided into two groups because we wanted to know which REM is important for memory consolidation. Is it immediately after the learning, or even the later part? So for this purpose, we did silencing in two type of mice. One, the first 6 hours of REM sleep, and the another is other later 6 hours of REM sleep. We found that silencing of adult one neuron indeed impair memory initial period, not into the later REM period. So upper panel is showing the percentage time freezing of the mouse when they receive food shock. Generally, they freeze into the same context, a kind of paradox for the memory. And you can see these mouse which were silenced during the REM sleep. So less freezing means their memory was degraded. Although the silencing of those adult bone neuron in the later period has a no effect. So from this study, we conclude that the activity of adult bone neuron during REM sleep is necessary for memory consolidation. Yeah, this is my last slide. I want to thank my mentor from All India Institute of Medical Science, Max Planck Institute, and I'm currently working in IIIS, those mentors who guided me through this process. Thank you very much for your attention.
Thank you very much, Dr. Dipendra Kumar. It is really very interesting talk on knowing the various aspects of REM sleep and why sleep is important for memory. And it's a thermoregulation aspects and hippocampal newborn neuron generation. So I invite audience, if they have any questions, could you unmute and ask any questions to Dr. Dipendra Kumar? We have 2 minutes as a discussion time. So please. Okay, I have a question about maybe how this during the REM sleep. You mentioned hippocampal newborn adult to newborn transition. Those neurons are actually activated. This process. So is it going like every 6 hours whenever REM sleep is ongoing, or it is in a everyday manner? Yeah. Could you give some specific?
This is very good question. Indeed, it is going almost all throughout 6 hour period. But as you know, the REM sleep amount is very short. The major portion of sleep is non-REM sleep. The REM sleep is around 10 to 25% of sleep is a REM sleep. So during that period of time, we have indeed seen the REM adult one neuron reactivation, which is very causal for memory consolidation.
Thank you very much. It is very insightful talk, and we thank you once again, Dr. Dipendra Kumar, for sharing your very latest and research on sleep. Thank you so much. And I now ask next speaker of our session, Dr. Donyang Lee, and he is from NAIST, National Institute of Advanced Industrial Science and Technology, Tsukuba, and title of is Protective Effects of Ashwagandha Extracts on the palmitic acid-induced lipid accumulation and oxidative stress in the hepatocytes. So please, Dr. Lee, could you share screen and start the presentation?
Okay, thank you for the introduction. So now I'm sharing my screen. Can you see it?
Yes, you are audible and we can see your screen. Please start.
Okay. Yeah. Okay. So hello everyone. My name is Donyang Li. As introduced, I'm a research fellow in AIST in Japan. Today I will introduce my research, the protective effects of ashwagandha extracts on palmitic acid induced lipid accumulation and oxidative stress in hepatocytes. First, is the background. Ashwagandha is a popular Ayurvedic herb trusted for a variety of health benefits in Indian traditional home medicine system, and it is enriched in steroidal lactones that have been shown to be the bioactives. Among the active methanolides, methamphetamine A has been shown to possess good anti-cancer activity, and methanol has been shown to possess anti-stress and anti-aging potentials, including improvement of memory and brain function. So in my study, they investigated the effect of ashwagandha extracts on steatosis and oxidative stress induced by the free fatty acid within a cell or organ that often affects liver as non-alcoholic fatty liver disease. Next is the results. We prepared extracts from ashwagandha leaves and stems, and analyzed their methanolite profile by HPLC. We can see they have different ratio and different concentration of methamphetamine A and methanol, and the cytotoxicity of the extracts was examined by MTT assay based cell viability assays. First, we examine the effect of one kind of saturated fatty acid called palmitate acid on hepatocytes who7 by treating them with various doses of palmitate acid for 24 hours. For the MTT assay result, we found that palmitate acid reduced the cell viability in a dose-dependent manner, and the non-toxic dose is around 100 micromolar. The intracellular triglyceride levels were elevated in non-toxic doses of palmitate acid, and according to the results of oil red O, the lipid can be stained into red color. The intracellular lipids increase in palmitate acid treated cells. And in order to investigate the protective effect of ashwagandha extracts, the hepatocytes were pre-treated with these 4 cans of ashwagandha leaf and stems extracts for 24 hours, and after that, treat the cell with palmitate acid for 24 hours. And for the oil red staining, we found that pre-treatment of these 4 cans of ashwagandha extracts, the lipid accumulation decreased. In order to find the molecular mechanism, first we detected the lipogenesis pathway. First, I would like to introduce you the lipogenesis pathway inside hepatocytes. So the free fatty acids are delivered into the cytoplasm by fatty acid translocase, which called CD36, and PPR gamma is a transcription factor, which is related to lipid uptake and also lipogenesis. The expression of PPR gamma is increased in response to saturated fatty acids, and it leads to the increasing of hepatic lipogenesis. And the fatty acids are oxidized in mitochondria mitochondria, which is controlled by PPR gamma, PPR alpha, and also in this oxidation process, reactive oxygen species will be produced, which will lead to the oxidative stress in cell. The products from the fatty acid oxidation also can be the materials for de novo lipogenesis. And the SRBP1C is a transcription factor that can stimulate the expression of genes related to de novo lipogenesis. The fatty acids from the de novo lipogenesis are materials for the triglyceride synthesis. First, the expression of PPR gamma and its target gene CD36 decreased. The expression of these genes decreased by the pretreatment of ashwagandha extracts L1, L2, and S1, S2 at the same time, and so these extracts decrease the lipid uptake and the lipogenesis to downregulate lipid accumulation. Next, we detected the lipids de novo lipogenesis pathway. So SRBP1C is a transcription factor related to lipogenesis, and we found SRBP1C and its target gene ACC1 showed decreased expression in the ashwagandha extracts pretreated cells, and its target gene fatty acid synthesis decreased expression in L1, L2, S1 pretreated cells, and this extracts decreased de novo lipogenesis to decrease lipid accumulation in hepatocytes. Next, we detected the reactive oxygen species production in hepatocytes. And the ROS can be produced during lipid oxidation, which can lead to oxidative stress. So we found that palmitic acid induced the ROS production, induced the ROS accumulation comparing with control group, and with ashwagandha extracts pretreatment, the ROS accumulation decreased. And we detected the lipids beta oxidation pathway. PPR gamma alpha is a beta oxidation related transcription factor, and the palmitate acid treatment increased the PPR alpha expression, which induced by fatty acid oxidation related to a beneficial effect in preventing lipotoxicity, and also provides more citrate for de novo lipogenesis. And we found that ashwagandha extracts L1, L2, and S1 decreased expression of PPR alpha, and also other extracts, the full cans of extracts decreased its target gene UCP2. So as a summary, we found that ashwagandha extracts decreased protect the hepatocytes from free fatty acids, palmitate acid induced lipid accumulation, by down regulation of lipid uptake and lipogenesis related transcription factor PPAR gamma, and also downregulation of the novel lipogenesis pathway by decreasing the expression of SREBP1C and its target genes. And also the extracts decreased lipid oxidation, decreased the free fatty acid induced lipid oxidation, and also ROS accumulation in hepatocytes. Thus, the ashwagandha bioactives are promising for liver health and non-alcoholic fatty liver disease prevention. So that's my research. Thank you for your attention, and thank you for giving me this opportunity to present in ISAJ Symposium. Thank you very much.
Thank you very much, Dr. Lee, for sharing latest research on the properties of ashwagandha and its effects on neutralizing the ROS in the hepatocytes. So now the session is open for answer to Dr. Lee. If the audience have any questions, please unmute and ask her. So yeah, you use the ashwagandha extracts and
Yes.
have its effect on transcriptional factor related to ROS regulation, which is PPAR alpha gamma. So what biomolecules from the extract, the property are the protein molecules or some other molecules? Do you have any specific molecular hint? Or is it theoretic? The extract contains a lot of things.
Yeah, yes. I also detected other molecules involved in lipid oxidation. Not only PPAR alpha. So,
Have you refined what kind of molecule within the extract it is? Is there a specific molecule that do this function?
yes. Because long-chain lipids have to be pre-oxidation in the cytoplasm first. So I detected the molecules involved in the pre-oxidation in the cytoplasm.
Okay. Thank you very much for sharing your research once again. And
thank you.
Thank you so much. Now we invite our next speaker. Could you please unshare the screen? Yeah, thank you. And next speaker is Dr. Zia Wang. The title of her talk is Ashwagandha Withanolides and hence muscle cell differentiation. And she belongs to National Institutes of Advanced Industrial Science and Technology, Tsukuba. So I ask Dr. Wang again, please share the screen and start the presentation. Dr. Wang, could you?
I'm sorry, I'm sharing my PPT now.
We can see you, and yeah, please share the screen.
I'm sorry, there's some setting problem.
Yu, and the title of talk is Cancer Targeted Delivery of Vitaferin A using Nanostructured Covalent Organic Framework. And Dr. Yu is from the Laboratory for Animal Science, Chinese Academy of Medical Sciences and Comparative Medicine Center, Peking University, and also from Biomedical Research Institutes and AST. So, Dr. Yu, could you please share and start your presentation?
Okay, thanks for your introduction. So can you see my slides right now?
Please.
Okay, and is the connection stable?
Oh, well, we can see your screen. Please start.
Okay, so good morning, everyone. So my name is Yu from the Biomedical Research Institute of AST. And thanks for the opportunity to let me introduce my work here. And today I will talk about the cancer-targeted delivery of vitamin A using a nanostructured covalent organic framework. So, generally, there are three major approaches in the cancer treatment in the clinics. But however, either of them has its own problems. And among the three, the chemotherapy is still the most frequently used approach that often accompanied with the other two. But there are still some major challenges in the chemotherapies, and one of the biggest problems is the non-targeted drug diffusion. Like for example, after the administration, the drug molecules invades to the normal tissues rather than the tumor section, and this process inevitably leading to a high toxicity to the patients. And for this reason, and all the cancer drug researchers are aiming to achieve targeted therapy. Like people want to find a drug that can specifically kill the cancer cells, but without harming to the normal cells. And to achieve this, I'm also proposing potential solutions by integrating the cancer drugs, nanomaterials, and the biomolecule together to generate biohybrid nanomedicines. And this nanomedicine could be potentially precise advantages such as high specificity, improved solubility, and controlled drug release. But however, the ultimate goal for this nanomedicine is to concentrate drug molecules to the tumor section and thus lower the toxicities. And for this purpose, we utilize the COF, which is a new class of organic polymers in the drug delivery system. And these materials could be synthesized from the two precursors, and the selection of the precursors is very flexible, and you can choose select them depends on your desired functionalities. And after the synthesis, the two precursors can link together to give a single ring unit, and this ring could be extended and stacking together to give a multi-layer structure like this. And the biggest advantage of this material is the high surface area and the large pore size. And these features enable the COF with an exceptional ultra-high drug loading capacity, which is much better than the other materials. So in this study, we prepared a COF structure with a pore size around 3.26 nanometers. And the hydrophilic part in the skeleton can offer a good water dispersibility of this nanocarrier. And on the other hand, the hydrophobic part will interact well with the loaded drugs. And we also conjugate folic acid, which is a cancer targeting ligand, on the edges of this nano carrier, and we examined the size of this nanoconjugates, and we found FA conjugation increased the size to 140 nanometers. And we also observe a very clear peak in the FA COF nanodispersions, which further indicate the successful conjugations. And the reason for us to choose the folic acid as the targeting ligand is because its receptor is folate is always present in the most of cancer cells rather than the normal cells. So theoretically, FA conjugates should be internalized more in the cancer cells. So to test this point, we labeled these nanoconjugates with nailrite, which is a fluorescein, and incubate with folate receptor negative and positive cells. And as our expectation, we found an increased cellular uptake by the positive cells, indicating the targeting effect. And for the next step, we were trying to encapsulate drug payload into the pore channel of this material. We select several chemo drugs of which size is smaller than the channel size, and we calculate the drug loading capacity, and we found that vitamin A has the best performance with a loading capacity around 44%. And after getting the drug loading nanocomplex, we were trying to investigate the cancer killing effect. And here we utilized the FA non-FA conjugated nanocomplex as a negative control, and we used after encapsulation with the drugs, we treated them with the cancer cells, FR negative and positive cells. And as you've seen on the right, in the negative cells, the cell viability doesn't show any difference regardless of the FA conjugation. However, in the HeLa FR positive cells, FA conjugation enhanced the cancer killing activity of vitamin A significantly, and we also performed the apoptosis assay and found here the FA COF loaded vitamin A induced a stronger apoptosis induction in the cancer cells, and this result was also confirmed by the Western blotting of apoptosis marker proteins such as cleaved PARP1 and cleaved caspase-3. And the in vivo performance of the anti-cancer performance of this nanocarrier was also investigated by injecting the nanocomplex into a mice which is bearing a tumor, FR positive tumor xenograft. And after the administration of the nanocomplex around 1 month, and we found here, as compared to the drug alone, FA COF delivery significantly suppressed the tumor formation during the treatment. And here, the immunohistological staining of the tumor tissue also indicates a stronger caspase-3 induction in the FA COF delivery group. And this data suggests that this nanocarrier can enhance the efficacy of loaded drug for selectively killing of the FR positive tumors in vivo. And it is known that many cancer drugs has the problems of the hepatotoxicity, and S2 and ST and LT is two major indicator for the liver damage. And we also perform the blood test to check the expression of these two indicators after our treatment. And as you see here, the FA cough nanocomplex doesn't induce any liver damage in the mice. But most importantly, we found this nanocarrier can also improve the inherent toxicity of the vitafilin A. So it reverses the expression of the AST and the ALT, which is induced by vitafilin A. And for the next step, we confirm the biosafety of this nanocarrier by the HE staining of the major organs after the treatment around 20 days. And we, from the results, and we found here, the FA cough doesn't show any difference with the PBS control. And similarly, body weight was also steadily increased during the treatment. So we conclude that these nanomaterials could be a possibly biocompatible drug nanocarrier in the future. And next is the summary. So generally speaking, we fabricated a FA folic acid conjugated nanocarrier, which can specifically bind into the folic receptor on the cancer cell surface, and thus enhance the cell internalization to release the drug in the targeted cancer cells. And that's it. Thank you. And if you have any questions in the future, you can send me email to this address. Thank you.
Very much, Dr. Yuri, on explaining your research, interesting research on this new molecular approach to give an efficient drug delivery. So the session is open for questions, and if audience has questions, please unmute and you can ask Dr. Yu. So this has a specificity to folate receptor and the folate. So receptors, is there a specific domain this molecule can recognize on these receptors? Because there are a lot of receptors on the, particularly ovarian and breast cancer you studied. So is it some CXCR5 receptors? What are those receptors especially it can recognize?
Yeah, do you mean like can we replace the folic acid to the other receptor ligands?
This particular modification on your molecule, yellow.
Yellow, yes.
Specific receptor on cancer cells, or it is general for so many receptors?
It the receptor for this folic acid is overexpressed in the cancer cells. It means that normal cells also has this receptor, but the expression is more in the cancer.
Is there a specific name of any receptor, would you say, or there in general many?
Yeah, it is specific for the cancer.
Okay, so thank you, I understand.
Okay.
Thank you very much again to you for interesting talk. And now we would like to move again, invite Dr. Zia Wine. Could you start your presentation again? If you are sharing screen problem is resolved.
Yes, I'm sharing now. Can you see my slides?
We can see, and please we can continue. Could you make a full view of PowerPoint?
Yes, okay. Is it full screen?
Thank you. Please.
Okay, I'm sorry for the inconvenience. Good morning, everyone. My name is Wang Jia. I'm a research fellow coming from AST. Today, I would like to share one topic with you. The title is ashwagandha vitalized enhanced muscle cell differentiation. Ashwagandha, also known as Indian ginseng, is an important medicinal plant that has been commonly used for its health promoting effects for over 3,000 years. It has been demonstrated that vitamin A and vithanol are active ingredients of ashwagandha extract prepared from root, stem, and leaves. Several studies have demonstrated the clinical efficacy of ashwagandha extract for the management of muscles. Volunteers in taking ashwagandha extract showed a significant increase in both muscle size and strength. Furthermore, a reduction in exercise-induced muscle damage was also observed. This study suggested the potential of ashwagandha as a supplement or medicine, but the molecular studies in muscle differentiation still need to be demonstrated. So next, skeletal muscle is the largest tissue of the human body and plays an important role in facilitating the motion and modulating the metabolite levels. When as a result of a disease or even a physiological process such as the aging, our muscles do not work properly. We become weak and frail and may even die. So hence, physiological structure and functions of skeletal muscles are important for the maintaining the homeostasis. Mature skeletal muscles are composed of multinucleated muscle fibers called the myotubes. Under the normal circumstance, an adult human processes skeletal muscle as a stable tissue with the considerable regeneration ability, which is performed by the skeletal muscle satellite cells. So, what is the skeletal muscle regeneration? Usually, the satellite cells are quiescent. Upon exposure to appropriate stimuli, like muscle injury, quiescent satellite cells are activate and undergo muscle cell differentiation process, which can be characterized by expression of muscle-specific proteins, withdrawal of the cells from the cell cycle and their fusion into multinucleated cells, which is called the myotube, to repair the injured skeletal muscles. A disorder of this repair ability usually occurs in various pathological conditions, such as the cancer, Parkinson's disease, or even the obesity and aging. Impairment of the skeletal muscle regeneration has a negative effect on a patient's life quality and increase their mortality, making the muscle differentiation an important target for the treatment. In this study, we investigated the effect of ashwagandha extract and its major vitalized on muscle cell differentiation using the C2C10 myoblast, which is a well-established in vitro model. So C2C10 myoblast were subject to the differentiation by culturing in media supplemented with either 2% house serum or 10% FBIs. As mentioned just now, the C2C12 the myoblast is commonly used for muscle cell differentiation. The cells grow rapidly under high serum conditions and transit to differentiation process after starvation or switching to a low serum culture. According to the morphology of observation under a microscope showing here, we can see that the cells cultured in differentiation media showed the differentiated states, including elongated and widened cylinder shape and multi-nucleated myotube on the fourth day, and kept differentiation in next 6 days. But the cells cultured in growth media did not show differentiation in the whole culture. Then we performed the cloning by serial dilution and select one clone, and did not show differentiation in growth media. However, showed slow and somehow uniform differentiation in the differentiation media for the study. We next subject the cells from this clone to differentiation in either control 2% house media or the test media supplemented with the vitamin A or vitanol. As expected, the poor differentiation was observed in 8 days old control culture. Meanwhile, the vitamin A cultures showed marked differentiation, which did not show much further change during next 2 days. And the vitanol treated culture showed much stronger differentiation. According to the previous studies, the content of the vitamin A and the vitanol has been shown to vary in different parts of the ashwagandha plant. We generated a extract from ashwagandha leaf and stem using an established method, and the extract was analyzed for the content of vitamin A and the vitanol by HPLC. We found that the extract 6, 10, and 12 obtained from stem contained relative low content of the major vitanolides. Vitamin A plus vitanol only 0.05 to 0.5 macromolar, and the high ratio of the vitanol and the vitamin A is 3 to 5, as compared with other extracts. Next, the C2C12 cells were treated with either differentiation media or differentiation media supplement with non-toxic doses with vitamin A and vitanol and ashwagandha extract by Western blotting analysis for measuring protein, which is a muscle-specific transcription factor involved in skeletal muscle development and is essential for the proper muscle differentiation. We found that the vitanol and extract 6, 10, and 12 they caused a higher reduction of myelin expression than the rest. In agreement with the stronger differentiation obtained by immunostaining for myelin expression in cells treated with these samples. This data demonstrated that the treatment of C2C12 cells with the extract containing a lower amount of total vitanolides and a higher ratio of vitanol, in particular, promoted their differentiation to myotubes. Protein aggregation and the accumulation of the molecular garbage is one of the causes of aggregated decline in differentiation capacity, and is closely related to neurodegenerative diseases. Therefore, we also examined if ashwagandha extract could recover or reverse some of the such damage by using two model AC systems. One is heavy metal induced aggregation of GFP protein, and another is the heat induced folding of luciferase protein. As shown in figure A, heavy metal caused the aggregation of GFP. Cells treated with vitanon and the extract that contained relative high content of vitanon showed maximum deaggregation. And this result matched with the differentiation potential of the extract. Quantitative measure of the luciferase activity in cells subject to the heat shock showing in figure C also revealed heat-induced misfolding and aggregation of the luciferase protein. In contrast to the GFP aggregation AC, luciferase activity was increased more significantly in extract 3, 7, and 11 that possessed relatively high level of the beta-ferrin A treated cells. This data demonstrated that the ashwagandha extract protected cells against the stress-induced aggregation of the proteins. As we know that the differentiation of the meioblast requires the functional degradative systems, including the autophagy to facilitate the elimination of the pre-existing structure and proteins in order to assist in formation of the multi-nucleated terminally differentiated myotubes. In view of this, we investigated the effect of ashwagandha extract on autophagy marker LC3B2. Western blot analysis here of the control and the treated cells revealed the increase in LC3B2 expression, especially in the cells treated with the extract 10 and 12. And this result was also confirmed by the immunostaining, suggesting that the ashwagandha extract could promote the muscle differentiation by regulating the autophagy. So last is the summary. Taking together, we can get the conclusion that the vitanon and vitanon-rich ashwagandha extract, they could cause the stronger differentiation and deaggregation and of the mito-induced aggregated proteins, and also activation of the autophagy pathways, also suggesting that the potential of the ashwagandha vitanon lies for the management of the muscle repair and activity. At last, I would like to thank all my lab members, and thanks for giving me the chance to present at this symposium. Thanks for your attention.
Thank you very much, Dr. Wang, for giving nice presentation. And I think we are almost having 1 minute. So if there is a quick question, we can accommodate from audience. Could you unmute if you have questions? Or you can directly send her through chat because of time limits. So we will move to the next talk now. Thank you, Dr. Wang, again. Please unshare your screen.
Yes.
And we have speaker, Dr. Rahul Mahadev Shetke from the Division of Applied Science, Life Sciences, Plant Molecular Biology, and Biotechnology Research Center from the Gyeongsang National University, Republic of Korea. And title of this talk is CRISPR-based genome editing tools for basic and applied biological research. So I hand over to Dr. Mahadev Shetke. Please share your screen. Yeah, we can see your screen. Please start. You have 15 minutes. Yeah.
Thank you, Chair, for introducing me. And I would like to thank the organizing committee for this invitation to talk about my ongoing work in Korea. I completed my PhD from AME University, Masuyama, Japan, and then I moved to Korea for further work. So in my talk, I will cover the basic aspects of the CRISPR-Cas system, or what were the different approaches we use to follow for the introduction of mutagenesis and get the better traits in the plants. So in my talk, further I will describe the application of these tools in crop improvement and what we do in our lab in the Korea, and further the regulatory aspects. So coming to the introduction of this topic, that we all know that the mutations are the basis of evolution and genetic variations. So any natural mutation in the in the population causes the creation of novel alleles, and those novel alleles confers new traits. So in this way, from the human civilization era, from the beginning of the human civilization, we have selected the better crops having the better traits. And that time we had no technology or knowledge of selecting on the basis of genetics, and we had selected the plants using the mutant phenotypes. So to generate or to fasten this process of mutation, we have developed several tools from the late 19th century and early 20th century. So earlier radiation and chemicals were used to introduce the mutations in the population of any crop, and later some DNA-based technologies have evolved since the 1950s, and one of them is like a transgenic approach, which gained more attention, but due to certain regulatory aspects, further this technology had several limitations to commercialize the products generated using the transgenic approach. And in early 2010, the CRISPR-Cas technology was introduced, and this technology has an immense potential, and it's based on this technology, several tools have been evolved, and this is continued to grow further. So what are the advantages that we gain when we use the CRISPR-Cas compared to the other technologies, that is the conventional plant breeding, mutation breeding, or transgenics? So these three approaches take significant time and several generations to screen and get the better phenotype or plants that have no other side effects, and we need to go through several regulatory aspects. But in case of genome editing, we can develop a new crop variety without waiting for 7 or 8 years. We can do in 1 or 2 years. So these molecular scissors, that is CRISPR-Cas systems, based on the endogenous DNA repair machinery that has two main pathways, that is non-homologous end joining and homologous recombination. And this non-homologous end joining pathway is an error-prone pathway. So when we cut the DNA, it produces the double-stranded breaks, and during repair by this pathway, we get the insertion or deletion, that is the 4 genetic letters of 4 DNA letters, ATGC, and this deletion or insertion causes the knockout or knockup of these targeted genes. So among the three, this kind of mega nucleases, CRISPR-Cas has got more popularity because it has more user-friendly features, like easy to adopt and apply in the experiments. So this technology was initially invented by Emmanuel Charpentier and Jennifer Doudna, and in 2010 and 11, they published papers where they adopted this bacterial immune system for programmed editing. And this contains mainly 2 components, that is the CRISPR-associated endonuclease. It is basically an enzyme, and single guide RNA. It is RNA and having some scaffold. So this guide RNA, that is the 20 base pair region around, we can target using this 20 base pair to any region in the complex genome, and it just need a recognition motif, that is NGG. So these tools have got more attention and application in human or in plants, and particularly in plants. To apply this technology, we follow certain basic steps. That is, we first need to know the target gene that will give us some improved trait, and after getting the design, after knowing the trait, that gene, we can clone the different components of this CRISPR system and deliver into the plant cell by using different methodologies, and we can screen the plants after we get the edited or targeted mutagenesis lines. So one of the major advantage of using this CRISPR technology is as we all know that our genetic package, that is DNA, is present in the chromosomes, and these complex structures are made up from the ultimately from the DNA, and that codes for the RNA, and that leads to the protein production, which gives us which are the functional units in the all living organisms. So we can edit any of this process using CRISPR-based tools. By using the basic features of CRISPR, several effector molecules have been fused with this Cas9 or particularly Cas endonuclease, and that can perform several modifications at different stages of the central dogma. So one of the tools is base editor. It is more specific and precise that we can edit the genetic letters, that is DNA letters, from A to G or C to T, and recently C to G also have been evolved. And we can make these mutations or changes at DNA or even RNA level. And one of the pioneer in this work is David Liu. So, and for the transcription regulation, that is the any gene expression can be controlled using these two tools, that is transcriptional regulators and epigenetic modifiers. And the recent addition of the tool is the prime editor, where we can make any change, like any particular base can be changed to any other base. Of course, this needs further improvement, but it has shown the application in human cells that we can make a small changes in the DNA that will give us the important functions or the important traits. So when we think of applying this for the basic or applied research, particularly in plants, we can do the two types of studies, that is to know the functional aspects of any target DNA, or to develop the biotic or abiotic stress tolerant plants. So one of the case study is in case of plants, the one disease, that is fungal disease, is caused by the fungal pathogen is powdery mildew, and the susceptibility factor, that is MLO1 gene, it helps the fungal pathogen to invade the plant. So if we target this gene was targeted using regular CRISPR-Cas tool, and by making the mutations in this gene, the tolerant phenotype or tolerant plants were obtained. Another aspect we can, another case study is, we can study the basics of how particular gene is involved in particular stress. And here is one example, that is the NPR1, which is a transcription factor. By mutation of this transcription factor, we can see that the plants become more susceptible to the drought. And so this way we can understand the functions of several genes before using them or before targeting them by using different tools. So in my current lab, we work on both the aspects, that is the development of new tool or the application of design tools or newly developed tools in the plant biology. And in case of new tools, we have precision tools or targeted random mutagenesis to mimic the directed evolution in plants. So there are several tools to make the targeted random mutagenesis, so we can have the evolved proteins. And one of the tool is ROS-based, that is reactive oxygen species-based mutator, that we have recently adopted. And this tool was developed using the ROS production enzymes. By fusing with the Cas9 protein, we can target this enzyme to the specific locus in the genome, and we can make the mutations in that region. So that mutations will lead to the different protein mutants that will give the different functions. Another platform technology that we have recently optimized in our lab is the screening of new tools or the optimization of new tools using the heterologous promoters. That these promoters are functional in bacteria and plants also. So using the same optimized components, we can perform experiments in bacteria as well as plants. So this new tool is quite useful to check the functionality of any cloned components or any component that we want to use further in eukaryotes. And recently this paper is published, and this work is done by Divyajyoti now he is a postdoc in our lab. So basically, the fungal and viral pathogens are the major factors that we lose a lot of yield in the agriculture. So by targeting two causative genes that make the plant susceptible to these mutant plants were generated for the one gene named as PELO, and another is the previously I have shown that MLO1. So these mutations leads to the development of disease-tolerant plants. So it also shows the applicability of CRISPR tools in plants. So there are several aspects that people consider when we think of the regulation of this technology for application or the commercialization of developed products in different parts of the world. So as we know that conventional breeding products are not regulated, but the genome editing or the GMO crops are having stricter regulations. But in case of genome editing, if there is a plant which is having no foreign genetic material, that can be counted as a non-GMO. And in some countries, there are these plants or these products are not considered as a GMO and having the relaxed regulations for the commercialization of these traits. So as per we consider Japan, this ESDN1, if there is a small change in the DNA, this product is considered as non-GMO and non-regulated. And in some part of the world, still there is a debate going on about this technology, whether to be regulated strictly like GMO or allowed to commercialize without any GMO rule application. So these are some of the papers that we have recently published and some other groups that people can refer to study more about this technology. Particularly this one review that we published in Molecular Plant, we have summarized everything about the tools that we use in the plant biology or even in animal. So finally, I would like to acknowledge my professor in the current lab, Professor Jayan Kim, and my colleague Dibyajyoti, who performed some of these experiments. And yeah, I really would like to acknowledge my PhD supervisor, Professor Morita, and Professor Hayashi, where I worked as a postdoc for 3 years in the Proteoscience Center, AMA University, Japan. And thank you for listening. And any questions are welcome.
Thank you very much, Dr. Shelke. It's a wonderful talk on the genome editing tools using CRISPR-Cas9 tech. It's open now for the questions to Dr. Shelke from audience. If there are questions, please unmute and ask here, or you can also ask him. Okay, so yeah, it's good, and so many CRISPR modifications are there for crop improvement. And now you mentioned about the ROS modifier fused with Cas9. So what is the special inducing protein in this case in plants? Because if you assume ROS overproduction fused with Cas9, so ROS are not targeted to the sequence, right? If they have a property to diffuse freely, so there will be more targeting using particularly the ROS. I think this is some consideration, but other requirements are good.
Thank you. There are limitations to use the ROS-based DNA editors directly in plants. Even though we have these enzymes that are inducible enzymes, still we observed some off-target in, at least in yeast, that system is optimized. Definitely, we need to have stricter control over the diffusion of ROS or the production of ROS. Yeah, thank you.
Hello, may I ask a question?
Yeah, please, yeah.
Good afternoon. Thank you for a very wonderful talk. I was very surprised to know that with this plant mutation, we can expected results can be achieved. But I was wondering with this, what happens to the plant after this desired mutation? Are there any undesirable side effects on that plant or after what we consume than the usual plant? Can you just pull my curiosity a little bit?
Thank you for the question. Yeah. Actually, when we use the GMO, we have to overexpress a gene continuously and constitutively. But in case of this CRISPR-based technology, after we cut the DNA of particularly we say gene, and after the ligation of that error-prone ligation of that gene, we say that like gene is knockout, or then we don't need that overexpression machinery. And for the after we get the plants, we do the crossing and segregation of the genetic material. That overexpression machinery is already we can select which is absent in the plant. So there is no any transgene in this technology, which is a major hurdle when we consider of GMO. So ultimately there will be no effect in the produced food material. Just the concern is the contamination of genetic material in the gene
Okay.
pool. Yeah.
So this is only true for the CRISPR, that there are no side effects. But as you mentioned, this technology is better than the other one. So does the other one have some side effects?
As a scientist, I believe like when we think of GMO, generally we should not have any fear. But
Okay.
as we overexpress certain foreign proteins which are not endogenous to the plant, some people may have fear like it may cause some allergy or something. But generally, these are easily degradable and digestible proteins mostly. So yeah, just to not have any issue, yeah, CRISPR is better to use
Okay.
than
So
GMO. Yeah.
safe to consume, I guess.
Yeah.
Thank you. Thank you very
Thank
much.
you. Thank you, Dr.
So thank you once again, Dr. Sharkey. And
Yeah, thank
now
you, sir.
we finish this session, and it's a wonderful session. So many invited speakers, and they have shared their very recent insights on the life science development and research. So see you in the next session, 3B, after the lunch session. So please join on this link, either room A or B, and please join, and we look for your active participation in the following sessions. So thank you so much. in-person participants for joining the 12th ISAJ Symposium. And this is your session 3B, and this is a specific session for young researchers, and in which a lot of particularly students will share their research. And this is a 5 minutes presentation time for each speaker, including 3 minutes of presentation and 2 minutes of question and answer. So please, all the presenters, keep your presentation in time so that you may also have the opportunity to receive questions from the audience. And I want to introduce the session chair for this session. 3B is Dr. Aga Fulke from National Institute of Oceanography, Mumbai, India. So I now request Dr. Aga Fulke to start the session and invite the first speaker.
Yeah.
Dr. Fulke.
Yeah, thank you, Sir Dr. Santosh. So as all the participants who are going to present in the session 3B, they knows that they will be getting the 5 minutes. Out of this, 3 minutes will be for the presentation and 2 minutes for question and answer session. So very first, I'm going to invite. Hello.
Okay, let me also inform one more important announcement.
Yeah.
So this session will have a best poster presentation award, and so this is for the participants. So best poster will be selected. So maybe we have.
Yeah. So the first one, He Hui Fu from AIST, and he's going to talk on the microRNA 780 regulate CARF, a trace regulatory protein, differentially in telomerase positive and negative human cancer cells. Hey Fu, please, you can share your screen.
So is there Mr. Hefu from AIST? Is he already there in the session? Is he present? Can we confirm?
Sir, sorry. He said he's waiting that the host will be allowing him to join this meeting now. He's trying to connect, but
Okay. Sorry for this. We will allow shortly.
yes, thank you, sir.
Please wait a moment. Sorry, this is all settings. Just a minute.
Hello.
Yes. Welcome.
Yes. Thank you.
Yeah. Please start. Thank you.
I'm sorry, I'm not allowed to share the screen.
Yeah, understand. Thank you. AIST lab, and he'll be presenting on anti-stress and anti-aging activities in lipoic acid, experimental evidence.
Hello, can you hear me and see my slides?
Yes, very clear.
Hello, yes, yes, I will just. Okay, so should I start now? Hello,
Yes,
can I start now?
please.
Yes, okay. So, good afternoon. My name is Ashish Kaul, and currently I'm a PhD third year student in the University of Tsukuba. And the title of my presentation is Anti-Stress and Anti-Aging Activities in Lipoic Acid, the Experimental Evidence. So, first of all, for the introduction, lipoic acid is a naturally occurring antioxidant, and there are two forms of lipoic acid: the R form and the S form. The S form of lipoic acid is optical byproduct when lipoic acid is synthesized in the industry. The R form of lipoic acid is more easily available in the nature, and it is found in natural sources such as organ meats and mitochondria, where it increases the antioxidant levels and helps in the metabolism of mitochondria. So why am I checking the stress activity in lipoic acid? Stress or chronic stress is one of the major causes of cancer and also age-related diseases such as Alzheimer and Parkinson, and also it causes mitochondrial dysfunction and genomic instability, which ultimately leads to cancer. So to check for the anti-stress activities in R form and S form of lipoic acid, first of all, I did cell viability assay to determine the low non-toxic doses and found that the dose range from 5 to 200 micromolar was not toxic. So based on this data, I chose 100 micromolar as the dose for my future anti-stress experiments. To check for an anti-stress, first of all, I did protein deaggregation assay, and I used two models. First one is the metal stress model, and the second one is the heat stress model. For the metal stress model, cells were transfected with the green fluorescent with the plasmid, which encodes for green fluorescent protein. And when cells were stressed with sodium arsenate, there was protein aggregation. On the other hand, cells were transfected with the plasmid, which expresses luciferase enzyme, and this enzyme's luciferase activity can be measured when a substrate luciferase was added to it. So when cells are stressed, there is misfolding of luciferase. So for both these stresses, first I gave the stress, and then after that I checked that if these stresses can be recovered with the treatment of R and S form of lipoic acid. So coming to the data, as I said in the previous slide, when cells were stressed with sodium arsenate, there was aggregation of green fluorescent protein as seen over here. Whereas when cells were recovered in the R form and S form of lipoic acid, there was protein deaggregation. On the other hand, for the stress assay, when cells were not stressed, there was normal activity of luciferase, whereas when cells were stressed at 42 degrees Celsius, there was no activity of luciferase. And then when cells were recovered in R and S form of lipoic acid treated medium, there was luciferase activity. After that, I also checked for the recovery from oxidative stresses in the mitochondria induced by hydrogen peroxide. For this, I did two experiments. First one is the JC1 assay. This JC1 dye, for the JC1 assay, I used JC1 dye, which is of two colors, red and green color. When the dye is added to the cells, the dye stains the healthy mitochondria as red and the unhealthy mitochondria as green. On the other hand, and I also checked the production of ROS in the mitochondria, which can cause damage to DNA, cellular proteins, and nucleic acids. And these oxidative stresses are caused when there is an imbalance of production of reactive oxygen species in the mitochondria. So for the first, this is the data, the JC1 data and the ROS assay data. And as I said in the previous slide, when I stressed cells with hydrogen peroxide, there was a large number of green staining, which shows unhealthy mitochondria. And also for the ROS assay, also when cells were stressed with sodium with hydrogen peroxide, there was increased activity of ROS. Whereas when cells were recovered in the R form as form of lipoic acid, there was no iron healthy mitochondria in the JC1 assay, and also in the ROS assay, there was recovery from the ROS stress. So in a summary, the R form and S form of lipoic acid, the low doses are non-toxic, and the non-toxic doses of lipoic acid, the R form and S form, cause recovery from metal, heat, and H2O2 stresses. So lastly, I want to thank my lab members, my academic advisors, and to the organizers of ISAJ for giving me the opportunity to present my talk over here. Thank you very much.
Thank you, Ashish. So I invite all the participants to ask any question. Anyone, any question on this?
If not, maybe they can directly text through chat and directly ask him. And we have very limited time. For 5 minutes is presented, so please keep in touch. We will move to the next. Please.
Fine. Fine. So now I invite Miss Malika Khurana, and she will be presenting on antitrust activities in the hypoxia-inducing phytochemical ectoterisgenin from Dilab AIST lab. Yeah, she already shared. Yeah, please start.
Good afternoon, everybody. Can you hear me?
Yes.
Okay. So good afternoon, everybody. My name is Malika Khurana, and I'm a second-year master's student at the University of Tsukuba. Today, I will be presenting on the topic anti-stress activities of the hypoxia-inducing phytochemical tectorigenin. So, what is hypoxia? When there is less supply of oxygen in the cells, it's called hypoxia. So recently, hypoxia has been implicated in cancer and neurodegenerative diseases. HIF1 alpha is the primary protein messenger in the cells for hypoxia, and I thought that its manipulation could be used as a treatment strategy in many disorders. So to screen for hypoxia-inducing phytochemicals, I created HIF1 alpha driven luciferase reporter cells, and for that I transfected a plasmid vector containing gene encoding for luciferase protein and gene encoding for HRE. After transfection, healthy cell lines were cultured, and it was seen that in U2OS osteosarcoma cells, the transfection was highly successful. Therefore, I selected the U2OS HRE cell line and carried out a screening assay. So out of the phytochemical library, I could find that drug number 6, or CL6 as we call it, showed a significant dose-dependent HRE response, as we can see here. To confirm this response, at the protein level also, I carried out a Western blotting analysis, and as we can see here, the HIF1 alpha, which is the primary protein messenger in the cell for hypoxia response, was upregulated in a dose-dependent manner. Then I wanted to see the cell viability when the cells were treated with CL6. So as we can see here, that CL6 showed dose-dependent short-term viability in short-term assays, MTT assays. So 5, 10, 20. So as the dose increases, the viability decreases. And I also wanted to see the long-term cell viability. And I could find that at safe doses, 1 and 5 micromolar, CL6 was relatively non-toxic. After that, I went back to the literature and I did some digging, and I could find out that moderate amounts of hypoxia, cellular hypoxia, is known to be neuroprotective and anti-stress. It also participates in learning, memory, and cortical plasticity. Therefore, we investigated differentiation-inducing and anti-protein aggregation potentials of my phytochemical CL6 using brain-derived cells. So what I did, I performed 2 experiments. In the first experiment, what I did was I chose C6 rat glioblastoma cell lines and used 2 safe doses of CL6 and treated these cells for 20 days with the safe doses. As we can see here in the phase contrast microscopy, the morphology changed significantly. On immunostaining, I could find out that proteins like GFAP and PST95 were upregulated. So we concluded that CL6 induced astrocytic differentiation. Next, I carried out protein aggregation and deaggregation assay. So what I did was I took C6 cell line, same as the previous experiment, and we transfected it with a vector containing green fluorescent protein. Now, this green fluorescent protein is aggregated with protein aggregation when there is cell stress. So we applied a metal stress, sodium meta-arsenide, 20 micromolar, and then we saw if CL6 could induce protein deaggregation. So as we can see here, there is no fluorescence. But after adding stress in the GFP transfected cells, there was protein aggregation in some places. And as we can see here, CL6 showed time and dose-dependent protein deaggregation. So in 48 hours, 10 micromolar was the most protein deaggregation seen. At the end, I would like to tell you that CL6 is tectorigenin. It is an unmethylated isoflavone found in the rhizomes of leopard lily, Iris domestica. It is a flower. It flowers perennially and grows in the hillsides of East Asia. Some known activities are anti-inflammatory, antioxidant. It also regulates PPAR gamma and releases erythropoietin, which is a very strong antioxidant in neurons. As a summary, I would like to tell you that CL6 is a candidate phytochemical that can be recruited for manipulation of hypoxia and hypoxia-related stress and neurodegenerative disorders. Thank you so much. And I would also like to thank ISAJ Tokai University, AIST India Dialab, TLSI, University of Tsukuba, and MEXT for sponsoring my study. And not last but least, but my mentors, Dr. Kaul and Dr. Wadhwa, and my whole Dialab team. Thank you so much.
Thank you, Malika. Is there any question? Yes.
It's a very strict. So again, to the participants, please keep in time, and we may not have time for questions if you take 5 minutes for presentation. If you need questions in your presentation, please keep finishing in the 3 minutes. So thank you again. Please next.
So the next one is Ms. Anisha Motifasari from Dialab again, and she'll be presenting on the Motapari Plus novel multimodal anti-cancerous small molecule. Ms. Anisha.
Yes.
Yeah, please share your screen.
Hello everyone, my name is Anissa Novitasari. I am a third year PhD student in the University of Tsukuba. And on these occasions, I would like to deliver a presentation on my research as shown in the title, Mortapari Plus, a novel multimodal anti-cancer small molecules. As we know that cancer is the uncontrolled growth of abnormal cells in the body, and it is considered the second leading cause of death worldwide. There are several causes of cancer, such as heredity, UV radiation, and others. In cancer cells, these causes will trigger several response mechanisms, including the activation of tumor suppressor p53, which plays an essential role in preventing carcinogenesis by inducing cell cycle arrest or apoptosis. However, it has been reported that this protein might become inactive when interacting with the heat shock protein 70, called mortalin in the cancer cells. Mortalin is highly expressed and bind to the p53 in the cytoplasm and preventing the translocation of p53 into nucleus, hence the failure of tumor suppressor mechanism. Until now, there are several natural compounds known as inhibitor of p53 mortalin complex, but in my study, I'm focusing on synthetic compounds. To understand the potential activity of several synthetic compounds, we established a screening step to search for potential abrogator of p53 mortalin interaction using p53 wild-type cell line. After several cycles, we identified top 6 candidates. we had reported the first novel anti-cancer molecules called murtaparib and the second molecules called murtaparib plus. In this study, I start my analysis from the cytotoxicity assay in human colorectal cancer, breast cancer, and normal cells. I aim to study the potential activity of murtaparib plus in different cell lines with different purity status. As shown in the figure A, HCT116, the LD1, and MCF7 cells were responsive to the murtaparib plus, but only T47D that did not give any response to this molecule. And then as shown in the simulation here in the figure A, murtaparib plus could preventing the formation of 15 mortalin complex through the in vitro analysis. Shown in the figure B and C, 53 fraction in the mortalin complex decreased significantly after murtaparib plus treatment, regardless the purity status. The inhibition on mortalin and 15 interaction by murtaparib plus result in the decrease in the mortalin and increase in the p53 expression. The inhibition also enabled the translocation of p53 from cytoplasm to the nucleus in the HCT116, LD1, and MCF7. And then, interestingly, I found that murtaparib plus induced apoptosis only in the HCT116, the LD1, MCF7, but not in the T47D cells. And also, murtaparib plus induced cell cycle arrest in the HCT116, the LD1, and MCF7, but not in the T47D cells. And I have also confirmed that this molecule has the potential to inhibit PALP1 activity in the HCT116, the LD1, and MCF7 cells, and resulting in the DNA damage, and also cause metabolic stress in the T47D cells. So murtaparib plus has multiple mechanisms as a novel anti-cancer molecules in the multiple cell line. And then finally, I would like to express my great appreciation to my supervisor, Professor Ronu Watwa, and Dr. Sunil Kaul, and all of lab members. And also thank you for the ISAJ for giving me opportunity to present my research result in this symposium. Thank you for your attention.
Thank you, Anita. Is there any question? If anyone having question, they can text also in chat box. So considering the time, now we move to the next one. And the next speaker is, participant is Hui Zhang from University of Tsukuba, and will be presenting on cell-based screening of anti-trace natural compound selection of ashwagandha withandel leaves and their molecular mechanism. Hui Zhang, please share your screen.
Yes, can you see my slide?
Yes.
Okay. Good afternoon, everyone. Thank you for giving me the opportunity to share my research. And my name is Zhang Hua.
You can start your video too.
Sorry.
Your video is not visible.
Oh, sorry.
Yeah, no problem. No problem.
Okay, you can start. Yeah, fine.
Okay. Is it okay now?
Yes.
Yes. Go ahead. Yeah, please go ahead. Yeah.
Okay. Sorry for the inconvenience. My name is Hua Zhang. I'm a first-year PhD student of University of Tsukuba. Today, I would like to share my research, and the topic is about the cell-based screenings for anti-stress natural compound, selection of ashwagandha withandolide, and their molecular mechanisms. So, stress is an inevitable component of life which accelerates aging and elevates risk of diverse disease. And culture cells showed molecular stress responses similar to those occurring at the tissues and the organismic level, and therefore regarded as a useful model system to identify anti-stress reagents. And oxidative stress has been implicated and closely connected to most cellular stress responses. So based on the health benefits of natural compounds, we have attempted to undertake cell-based screening for natural compounds possessing antioxidative stress activity and resolve the molecular mechanisms of their anti-stress effects. So first, several round cell-based screening were operated, and cells were subjected to oxidative stress by exposure to paraquat, and then allowed it to recover in either medium or natural compound supplemented medium. The compound that resulted in protective effect, which was measured by cell viability assay, and the result is showing here, were selected for the second round screening. And in the second round screening, cadmium nitrate, heavy metal stress, and cobalt chloride, which induces hypoxia stress, were recruited. And both of these two stresses were reported to be related to the oxidative stress. Finally, several compounds that belongs to ashwagandha with lines or extract, as shown in this table, were selected for their outstanding performances of reducing cell death induced by these three kinds of stresses. And axis ROS release in oxidative stress targets macromolecules, including DNA and proteins, triggering cellular damage, which becomes particularly problematic during aging. So we also operated several related assays and the results revealed in ROS accumulation, and also DNA double-strand damage, and also protein aggregation in stressed cells, and was prevented to a large extent in cells treated with ashwagandha extracts and lines. We can hear in this the drug-treated groups showing weak fluorescence. Besides, we also found ashwagandha with lines treated brain-derived cells showed strong differentiation, as marked by the respective differentiated cell morphology. Here we can see the long neurites, and also the expression of neural differentiation related markers GFAP and neuron. So finally, a short summary. Taken together, the study strongly suggested anti-stress and pro-differentiation potential of ashwagandha with lines and extracts that may be recruited or management of stress and old age-related ailments. So finally, I would like to appreciate my supervisors and also the team members, and also thank you for your attention.
Thank you, Hui. I have one question. Somewhere you have presented cadmium, this heavy metal stress, and one is hypoxia for cobalt metal you use, right?
Yes.
So what was the control for that?
The control is first is like negative control, that cells didn't treat it with the stress, and then not positive control. Another control is the cells treated with stress but recovered in only medium, not drug supplemented medium.
Okay, very good, very good. Thank you. So is there any question, or shall we move for the next one? Dr. Santosh?
We move, and if there is a he who fails, he can finish this. Okay. He can be ready if he is.
Yeah, we can. So next participant is Mr. Vinod Kumar Jangam from IIT Banaras Hindu University, India, and he will be presenting on use of sequencing batch reactor for simultaneous carbon, nitrogen, phosphorus removal from domestic wastewater by alternating cyclic times. Mr. Vinod Kumar Jangam, are you there?
Yes, sir. Yes, sir.
Yeah, please share.
Sir, can you see my slide?
Yes, yes.
Okay, good afternoon, everyone. My name is Vinod Kumar. Currently, I'm a research scholar in IIT BHU. Today, I will be presenting.
View of your presentation mode. Yeah. On PPT.
Yes, sir.
Yeah, full view, slides, presentation mode of your. from the
Yes, sir.
Yes, presentation mode.
Okay. So today I will be presenting the use of sequencing batch reactor for simultaneous removal of organic carbon, nitrogen, and phosphorus from domestic wastewater by alternating cycle times. So these are the contents that I will be presenting. So generally, domestic wastewater, it will generate nitrogen concentration of around 26 to 70 milligram per liter and the phosphorus of 9 to 30 milligram per liter. So generally, the nitrogen in the wastewater, it will be present in the form of ammonia ion, nitrite, and nitrogen gas. These are all like inorganic compounds. Similarly, the total phosphorus in the wastewater, it will be present in the form of polyphosphate, orthophosphate, and organic phosphate. So if the discharging wastewater, it is having excessive nutrient concentration, then it will be cause the eutrophication and also the contamination of groundwater table in the excessive amount of area. So there are like different organizations, WHO and US EPA have given maximum limit of discharge for the different species of nutrient. Like phosphate should be less than 0.5 milligram per liter, nitrate should be less than 10 milligram per liter, ammonia nitrogen should be less than 5, and nitrate should be less than 0.1 milligram per liter. So my major objectives are like to design and study the sequencing batch reactor for the treatment of domestic wastewater from the individual houses in the Varanasi city, and also the characterize the municipal wastewater characteristics, and optimize the SBR operation for the different operational parameters to achieve efficient and sustainable treatment, and also to verify and validate the setup using the data from the Ramnagar sewage treatment plant. So generally, the sequencing batch reactor is a sequence process. It is consisting of different stages like filling phase, reactive phase, settle phase, decanting stage, and ideal stage. So during the reaction phase, there are all the metabolic reactions can be takes place, and also the segregation of solid and liquid can take place in a single reactor. So this is a more effective method for the removal of various contaminants like organic carbon, nitrogen, phosphorus, and suspended solids. So these are the characteristics of the municipal wastewater of Varanasi city. So this wastewater sample was collected from the outlet of grid chamber and analyzed for the different parameters like pH, total suspended solids, BOD, COD, total nitrogen, and phosphorus. If you see the characteristics, there is a small deviation in the phosphorus, total nitrogen, and phosphorus, whereas there is a little high deviation in the BOD, COD, and total suspended solids. So this is the experimental setup, which is having a diameter of around 9.4 centimeter and a height of 116 centimeter with a working volume of 8 liter. So it will be provided with the different ports for the different volume exchange ratio, and it is also provided with a diffused aeration system for the aeration. This setup was already fabricated and installed in the laboratory. So this is the operational procedure with anaerobic, oxic, and anoxic sequence batch process. So there are different phases are there. So in the first phase, it will be stabilizing for the microorganisms to optimize the conditions. And in the subsequent phases, there is an introduction of different phases, different stages like anaerobic, anoxic, by reducing the cycle time and hydraulic retention time. So coming to summary, my experimental setup is already installed in the laboratory, and the characteristics of the wastewater has been studied and also planned for the synthetic wastewater. So the future work will be the seeding of the reactor with the activated sludge from nearby treatment plant, and also the wastewater sample will be collecting at a regular interval and analyzing for the COD, BOD, ammonia, nitrogen, total nitrogen, and phosphorus with a standard method which is given by American Public Health Association. And also the monitoring of pH oxidation reduction and the potential will be carried out to control the nutrient removal during the treatment process. And also the statistical analysis will be applied to the each phase of the SBR so that the optimization of this COD, BOD ammonia can be obtained. So these are my contents. So that I would like to thank my supervisor, Dr. Mahendra Kumar Pal, for this opportunity. Thank you.
Thank you, Vinod.
Thank you, Mr. Vinod. Considering the time now, we'll move to the next one. And in between, I would like to announce the first participant. If he is available, be ready with his presentation. Mr. He Haifu. So the next one is Hitesh Supe from Hokkaido University, and he will be presenting on geospatial assessment of expanding solar farm for electrification of India. I request him to stick to the time for presentation. Mr. Hitesh Supe, are you there?
Yes, I'm here.
Yeah, please share.
Mr. Vinod.
So can you see my screen?
Yes.
Okay. Thank you for the introduction. Thank you for opportunity. So this is Hitesh Supe. I'm from Hokkaido University, and today I'm going to speak on geospatial assessment of expanding solar farm for electrification of India. Okay, so India is third largest producer and consumer of electricity in the world, and its daily demand for electricity is not met yet. As you can see, for February 17, the electricity supply was short by 723 megawatt, and it changes daily around. It can go as high as 1 gigawatt also, and now the country is trying to change its focus from thermal-based energy source to renewable energy source, and its more focus will be on solar energy sector, because this technology directly harness energy from the primary source, that is the sun. And there is a target set by the country for it to achieve its HDG goal of 450 gigawatt of renewable energy by 2030, and the major contribution will be from solar energy, that is 280 gigawatt. 450 gigawatt is even higher than the current total generation of electricity, which also includes coal-based source. So you can imagine how big the solar farms will be needed for, will be installed for this to be achieved. And there are also some more initiatives like One Sun, One World, One Grid project, which will connect many countries. So it's likely that the solar farm expansion will be rapid and it can have an encroachment on land. So the current share of renewable energy is 24%, and of solar power is around 10%, and it has a capacity of around 40 gigawatt. So, and it's also likely that the solar farm may increase on some water bodies on the coastal areas and seashores and all. So it can cover a major portion of the water also. And here are some examples of some floating solar power plants in India, that is in Telangana, Tamil Nadu, and some are in Andhra Pradesh. So the main question arises is whether these solar farms are really helpful in electrification of India or not. So for that, I plotted a map of all the solar farms in India, locating all the farms in India of last year. And you can see here the solar farms are located in northern region near UP, Bihar, and also in some parts in south in Andhra Pradesh, Maharashtra, Tamil Nadu. So one way to assess this is by looking at the nighttime light data of this region. So I took the nighttime light data of 3 years, that is 2014, 17, and 20, and made an RGB image of them. So that you can see the changes. So basically, wherever you see greenish and bluish tint is the area where the nighttime light has increased. So the most greenish bluish tint you can see in the north region from in UP, Bihar, and some part in Maharashtra a little bit, and in south. So this also visually it says like the nighttime light has increased in this region in past 6 years. And if we see if I take the average of all those states and see their readings, so then you can see that most of the states have increasing graph like this, and so all have a positive response. So in short, you can say that the solar farms are very effective in the rural electrification programs, and there are some statistics which supports this these results. So that's all. Thank you.
Thank you, Mr. Hitesupe. So considering the time, now we'll go for the next one. And the next is Jenyu Chen from the same Tokaido University, and presenting on the machine learning based approach of for assessment of typhoon induced forest damage in case of Taekwondo.
Hello, everyone. Hello, can you see my screen?
Yeah, please go ahead.
Hello, everyone. My name is Chen. I'm first year PhD student in Hokkaido University. So today my presentation title is Machine Learning Based Approaches for Assessment of Typhoon-Induced Forest Damage in the case of Hokkaido. So this is the background of this study. Typhoon is a major natural disaster affecting forest ecosystem in coastal area. So also in Japan, typhoon caused a lot of damage. So in 2016, have 3 typhoons hit Hokkaido. So our study area is located in eastern Hokkaido, so next to the Obihiro city, and the typhoon pass is nearby the study area. So here is the objective of this study. The main objective is to detect the change of forest cover damage by 2016 typhoon in the part of Hokkaido using geospatial data. And this is the methodology flowchart. You can see here is the image processing part, and here is the machine learning methods applied. And here also represent the topography parameter and also wind parameter extract and also analysis. So this is all in vegetation index what we used in this study, especially DVDI and also FVC change. So for vegetation index, we use the spatial methods to classify the damaged pixel or undamaged pixel. And also using Google Earth high resolution image for validation and build the machine learning data training base. This is an example to showing how we do the interpreting. So this is the damage map of this study area. The red part is the damage area where we estimate, and also for imaging is based on different vegetation index. So this is a damage map based on two machine learning classifiers result, which is SVM classifier and also RF classifier. So here is the overall accuracy of our study. So from here we can see the 2 machine learning methods give the highest accuracy value. This figure is showing the relationship between each parameter, the relationship between this each parameter and also damage pixels. So here's the conclusion. First is optical satellite image can be used to assess forest damage purpose study. The second one is machine learning method give the most accurate result. And the last one is for topography or wind parameter, we didn't find any linear relationship between them. So thank you for your attention. Yeah.
Thank you, Dinu. I have one question for you.
Yeah.
As you mentioned in your presentation that you have collected the data where the typhoon was damaged the forest, right?
Yeah. Yeah.
So I wanted to know like how you assess the damage in terms of the tree fall or like in terms of the death of animals or the other thing on the forest or any control you have taken or only the in silico analysis of the data collected at the time of the typhoon.
For typhoon data, we mainly just take the typhoon path, so we can see this is about typhoon path data.
Okay.
And also forest only focus on forest area. So
the past in the sense of for how much years back, like before typhoon, just
and just after typhoon.
Okay, Jinu, thank you so much.
Thank you.
So we'll move to the next one. Next is Miss Shweta Singh, and she'll be presenting on capturing and transmission of drug-resistant gene by conjugative plasmids. Meanwhile, Mr. Haifu can be ready with the presentation if he is there, or someone can just convey him to be ready with presentation. So for a while, Shweta Singh from Shizuoka University, capturing and transmission of drug-resistant gene by conjugative plasmid. Ms. Shweta, are you there?
Learning model. So Ms. Ravina Raj from Hokkaido University, please start.
Yes, can you hear me? Yes, can you hear me?
Yes, please start.
Yes, yes, can you see my PPT?
Yes, please start. Yes, we can see and listen. Please.
Yes. Please let me minimize this.
Please go for the yeah. Start.
Yes. Please excuse me. Yes. Good afternoon, everyone. My name is Ravina Raj. I'm PhD student in in Hokkaido University, and I'm going to present my research on the study of geomorphological changes in chamber ravines of India using Tandemix SAR and machine learning model. And so first of all, ravines and gullies are the most degraded type of landform, and ravines is the complex network of several of gullies, and gullies are the deep channel made by concentrated flow of water, and it removes the upland soil and parent material and make the land very unfertilized or unproductive. And this land degradation is a very big problem in front of world and also in India. Around 3.97 million hectares land is affected by ravines in India, and in which 2.7 million hectare, almost 70% is covered by chamber ravines only. And there are several of action and programs has been initiated by government and local community to reclaim the revenues land. And recently, central government with collaboration of World Bank has decided to convert a large area of ravines into land for can be used in agriculture. In Madhya Pradesh. So for this, I think gully erosion susceptibility and monitoring is very important to mitigate the gully erosion hazard risk. So the main objective of this study is the morphological changes assessment in jumble ravines of India, where by making gully erosion susceptibility map and estimation of gully volume changes with the help of remote sensing and machine learning model. So this is my study area. My study area lies in central of India, in Madhya Pradesh, in Bhind district, and this plot of my study area, which I have divided into 2-3 parts. For my machine to prepare my machine learning model, I divided in my study area in training area and its validation area and the left area, and this is the 2-year plot of 2012 and 2017. So the common area between 2012 and 2017 is training area and validation area, and the uncommon part I choose for my testing area. So in my methodology, basically I extracted DEM data from like dynamic SAR for year 2012 and 2017, and I processed the DEM with the help of NV SARscape. So like I said, with the help of common area between 2012 and 2017, I extract the volume change between the volume of gullies change between 2012 and 2017. I extracted from the DEM difference with the help of ArcGIS, and also I extract all the gully affecting factors with the help of GIS, Google Earth Engine, and Google Earth, and to make my model, my training model and validation
Ms.
model.
Ravina, you have 1 minute left.
Okay, I extracted all the value of the factors for my random points, and I made my training model and I validate it, and I fit this model on my testing area. And I got my gully erosion map and volume estimation map. These are all the map of gully affecting factors, and I also did a multi-collinearity check. And in machine learning, I tested 4 machine learning: random forest, logistic, naive based, and random tree. in which random I found random forest is the most accurate machine learning for this with the highest value of ROC, and this is my gully erosion map, in which I found this area is highly affected by gully erosion from very low to very high classification. I found 4.487.57 kilometers square of area is facing very low gully erosion, but 48.69 kilometers square of area is facing very high gully erosion. And this is my volume estimation map. Oh, sorry.
I'm sorry, we have to move next session. So please, yeah.
Yes, okay. Yes, so I'm finished almost, and it was my volume estimation map, and thank you very much.
Thank you, Mr.
Thank you, Ravina. We'll go for the next one, Shweta Singh.
Okay. Okay.
Shweta, please share your presentation.
Yes. See my
Yes,
poster.
yes. Please go ahead.
Sorry for the inconvenience.
Please, so we have 3 minutes for your presentation.
Yes. Good afternoon, everyone. Myself, Shweta Singh. I am a doctoral student at Shizuoka University. And first of all, I would like to thank organizing committee to giving me a chance to present my work. So the title of my work is Host Range of Ink and Plasmid with Antibiotic Resistance Gene with antibiotic. Oh, I'm so sorry. Can you able to see my poster?
Yes, yes.
Genes captured from environmental samples. Okay, so first of all, moving to the introduction part, the ink and plasmid is a self-transmissible plasmid and mostly found in enterobacteria of human and the animal. This plasmid has many resistance against antimicrobial classes such as ESBL, aminoglycosides, sulfonamides, tetracycline, and the streptomycin. So from the study, what we come to know, ink and plasmid were considered to show a broad host range, but it is not necessarily to clearly seen yet. So the objectives of my study is to investigation of the host range of the ink and plasmid. So for capturing the plasmid, we perform two methods such as triparental matting and the biparental method. In the triparental method, the plasmid from the environmental sample is transferred to the intermediate donor, and both the plasmid conjugate and finally transferred to the recipient strand. In biparental matting, what we did is plasmid from the environmental sample is transferred to the recipient cell. So by this both method, we collected total 5 ink and yes, hello. By this method, we collected. Are you able to?
Please continue. Yeah, we can.
Yes.
So please.
Yes. Yes. So we collected total 5 ink and plasmid. As we can see here, this is the name of the plasmid, and these all the plasmid has antibiotic resistance genes. So after collected the plasmid, we perform the filter matting assay to check the transmission of ink and plasmid to the other bacteria. So in this method, what we did is we are checking the transmission of ink and plasmid from donor to the recipient strand by using of fax technique. By using of fax techniques, we selected the trans conjugate And we are sorting the cell on the basis of green fluorescent protein. So after this, this is the result of filter matting assay. Here, what we saw that the the transmission of ink and plasmid can also be transferred to the beta proteobacteria, and it has never been reported before. And the most of the transconjugants were Ralltella erythrolytica. So coming to the conclusion, total we collected the ink and 5 plasmids from the river sediment of Tama River and the wastewater plant. And these all the ink and plasmid can be transferred to the not only gamma protobacteria, but also show the transmission or to the beta protobacteria. In future work, we are going to check the transfer frequency of ink and plasmid from donor cell to the recipient cells. I would like to thank my professor and my lab mates to their support. Thank you so much.
Thank you, Shweta Singh. Thank you so much. And next we have Mr. Hu Hing. So please start your presentation.
Yes, yes, yes. And sorry, the topic is microRNAs 708 regulates cough differently in telomerase positive and negative human cancer cells from AST. And the first telomerase play a major role in structure and functional organization of the chromosomes. And they did, in the normal cells, telomerase gradually shorten with each cell division. In cancer cells, the telomerase is be length by telomerase and or alternatively lengthening of telomerase. So it's achieved either by activation of telomerase or an alternative mechanism of lengthening of telomerase. We call it ALT. Distribution of either of this mechanism has been shown in induced DNA damage signaling, leading to senescence or apoptosis. So in this study, we aim to examine the differences in microRNA species between TAM, telomerase positive cells and the alt tumor cells in order to shed light on their roles in the two different telomerase lengths and maintenance mechanisms. So in order to narrow down the difference in micro expression pattern, so around 300 micro RNAs were either not expressed or not differentiated. So among this, we selected 9 candidates of microRNA. So that has at least five-fold difference in this expression between TEP and the alt cells. So microRNA 708 was further confirmed to be the predominantly overexpressed in alt cells in a large panel of isogenic and non-isogenic type and alt cells lines. So we selected the micro 708 as one of the micro RNA with high level of expression in alt cells for further studies.
1 minute more.
Yes. In order to determine if the cough is the target of 708, we perform a luciferase report assay so that analyze overexpress of 708 causes strong inhibition of cough, and the microRNA 708 target cough in both TEP and alt cells. Overexpress show 708 shows decrease in cough protein levels also. And the high level of microRNA is 708 responding low level of cough in all cells also. So next, we microRNA 708 differently regulate cough, ATM, and P53, P21 signaling in the TAP and all cells. So we can see, whereas ATR did not show different differential expression in RNA or RNA 708 overexpressing and compromise cells. So major cells the regulator in tumor cells through its roles as a regulator of P53 and P21 activities. So microRNA 708 overexpress cause an increase in ATM in hot cells also. So we demonstrate that the microRNA 708 is a tumor suppressor microRNA, and further, it suggests that the TEM and the odd cells may differ in their cough-mediated stress response, and hence should also be considered for the cancer therapies. Sorry for the convenience. Thank you for your attention.
Thank you very much, Mr. Ishu. And now, continue with our session 4B, which is again invited speakers, and I'm the chair, Santosh Gotwal. Welcome you all speakers in the session 4B of the 12th ISAJ Symposium. And our first speaker in this session is Dr. Kalpesh Patil from Jamstack. And the title of his talk is Long-Led Prediction of ENSO using Convolution Neural Networks. So, Dr. Patil, please start sharing your screen and presentation.
Yes. Is it slides are visible? Hello?
Yes, visible, and 10 minutes for presentation.
Okay.
Question answer well,
okay.
And so that you may have questions.
Okay. So thank you very much, Dr. Santosh, for introducing me. I thank this for this opportunity to Dr. Kedar Mahapatra sir for allowing me to share my latest research work based on El Nino Southern Oscillation prediction for the long leads using convolution neural network. I also thank the ISJ Symposium organizers. I would like to thank all the collaborators for their suggestions. I am a postdoctoral associate at Japan Agency for Marine Earth Science Technology at Yokohama office. So the outline is to introduction of an ENSO, then what are the past forecasting works, why we need deep learning for it, and the results of the proposed approach, and followed by summary conclusion and future works. The brief introduction of ENSO, it is called as El Nino Southern Oscillation, which is a climate recurring phenomenon appearing in the tropical Pacific waters in the central and eastern parts. We can see in this two right side figure. And the vast area of this tropical Pacific gets warm or cool than relative to the normal. And that creates a lot of changes, which we'll see in the next slides. And these changes are tracked along a small area, which is center of the central and eastern part called Nino 3.4. And the average surface temperature anomalies are plotted against the time, which is called as Oceanic Nino Index, to verify the different ENSO events. If the index is more than 0.5, we call it as warm event, which is El Nino. And the vice versa for the colder one is La Nina. And it is the periodicity of that event is said to be around 3 to 7 years. And it usually peaks in the month of December and decays in the March. And we can see in this figure, it has a very global impacts, like different parts of the world get warm or dry. And also it is like different countries have different precipitation because of this phenomenon. And this is because of the unusual warming and cooling of these two central areas in tropical Pacific. that leads to enhanced convection and reduced convection depending on the warm and cold episode. And thus it affects the local and global community. And the different impacts are like disease outbreak, agriculture yields, natural disasters, water resources, energy demands, fisheries, wildfire, and a lot of things. This ultimately impacts the economy. And that is why we should forecast what is going to happen in the next few months or like at least 1 year before. And there are different works which has already been targeted for doing so. We will see what methods they have used. The different methods can be subdivided into two major groups. The first is state-of-the-art dynamical models. They are based on the balance of mass and energy equations. These are a couple of examples from that, and the syntax scale interaction frontier science experiment is from our laboratory, application laboratory, which is doing very well in the seasonal predictions, I would say. These are some issues pertaining to such approach. First is spring predictability barrier, which means during the spring season, the skills drop rapidly. Their long lead skills are very poor. Perhaps they are complex and not identify the precursor. What are the precursors? That also we will see. And on the other hand, this data-driven methods are less assumptive techniques. We can see these simple techniques can be used in the past. But the issue is that we cannot go for the longer lead time. The predictability is limited only for the medium range. And we cannot use the full map of the inputs. We have to average it around that region. And precursors are not possible to find out. Therefore, the interpretations are difficult. But in latest deep learning, these kinds of issues are not more. But still, their poor predictability of long lead is a concern. And there is a lot of change in the skills for the different seasons. Now we see why deep learning. Because we can give the complete map of area as an input, so we can account for the spatio-temporal variability easily. We don't need to average for the important region as shown. And also we can interpret, for you can see in the example that when the dog and cat is being classified, we can see the face is giving more input. Similarly, like in pneumonia detection, that's why the interpretations are easy. CNN is one of the best choice among the other algorithms. We can track the different features, important ones. And such has been used in the past for a lot of research in oceanography, climate, and meteorology. And before the proposed approach, I would like to introduce three major phases of ENSO event. First is normal, where the western is warm and the eastern is cool. And this subsurface temperature profile is called as thermocline, which is shallow in the eastern and deeper in the western for the normal condition. But what happens to the La Nina condition, which is the cool episode of this event? This thermocline goes much up than the normal condition, and both the regions are very cool. And in the warm region, both the regions are warm, and the thermocline is in much deeper region. And to account for the different inputs, the first input I am giving is sea surface temperature anomaly map for the global ocean. Second input is the average of subsurface temperature from 0 to 300 meter. And the other input is like different maps at different depths. And this is how I devised two different experiments based on this, different major phases of ENSO to capture. And I have targeted for the longer lead around 23 months, around 2 years, from 1.5 years to 2 years. And this is my proposed approach. Different maps for the past 3 monthly inputs are stacked together. They have been convoluted, and then different layers are followed for the feature extraction and the feature resizing. And the one constituent block is called as convolution block. Such several blocks were applied. And finally, we use the traditional methods to connect it to predict the seasonal ONI index for particular season. And I use 12 different models for 12 different seasons. And different hyperparameters, which are shown in yellow, are optimized by a random search algorithm. And coming to the results, first, the analysis of results is based on this parameter called correlation scale. It measures the direction, whether the predictions and the observations are in phase, out phase. If it is very close to one, it suggests a very good agreement. And the root mean square error. It measures the distance between the predictions and observations. The smaller is better. And the inputs, what I used for the training CNN is from Kobe and Soda data, and testing is done from the other data to consider not the biasness. And the resolution is around 5 by 5 degrees, little coarser. Training period is somewhere around 101 years, and testing is to the recent 34 years. Now we see at the skills of proposed method for first experiment, vertically average temperature, through different seasons and through different lead time. We can see the proposed model is doing pretty good for the higher lead time, and this hatching shows that this correlation skill is more than 0.5, which can say 50% of the times the direction is right predicted. And more darker is more better, more lighter it is more worse the model. And you can see in many of the boxes we can find the scale is more than 50%. Perhaps for some seasons during the summer and the early winter, this scale is a little bit dropped. And when I compared these predictions from the dynamical prediction systems and for the latest deep learning study, which is doing the similar kind of predictions, I found that the proposed approach is better for doing in both of the ways. For all the lead times plus different season, there is no drop in the accuracy. Plus, we can see that after spring season, there is a drop of accuracy and which has never regained again properly. And that is what is called a spring predictability barrier. Perhaps in the proposed approach, the results are coming back. And in the second experiment, what I found is the results are even much better than the first one. And there is no such spring predictability barrier observed for the early lead times. And late lead times, or what is called as longer lead times, are even better than the earlier approach. So we can say that this proposed model is good for crossing the spring predictability barrier, plus the long lead skills are really good. And also it captures the variation of the ENSO events in different seasons properly. Now, this event peaks in the December. So this is the comparison of the ENSO events in the December against the observations. And we can see the three curves. The orange one is from the dynamical model, green one is from the first experiment, and the blue one is second, which we can observe that the blue one is standing clearly out with the correlation is matching with more than 75% of the times during those testing period. In the recent paper, it has said that the skill is dropping for the latest century than the earliest one. So after 2000, it has been observed in the many models that there was a drop in the skill. So to check whether these kind of proposed models are sensitive towards a change or not, I did analyze the accuracy for 3 different periods. The gray one is for the total period of 34 years. The first blue one is for the early 17 years, and the orange one is the later 17 years, which is the latest century, the past 2 decades. And we can see that there is a drop in the first of the dynamical models and then the first proposed approach. But perhaps such drop is not visible in the second proposed model, which means that the change in dynamics, what is happening from in the latest period, the model is able to capture very nicely. And as I mentioned that these things should be interpreted. Now we can see what are the constituting areas which are responsible for such good skills, as we have seen in example from the dog and cat perspective and the pneumonia detection perspective. This is for the 1998 event prediction. For example, we can see this peak. So this peak we're talking about right now. And we can see the different areas which are contributing for such good skills. For example, the southern Indian Ocean, the western part of the tropical Pacific, plus this western hemisphere pool, subtropical Atlantic regions are contributing much better. The darker is more contribution and the lighter is lesser contribution. And such similar things were also observed in the second experiment, which is the subsurface temperature experiment. And in some analytical studies, exactly these regions have been found as a trigger of the ENSO events in future. So perhaps the study is properly analyzing and interpreting that the physics of the ENSO events, not completely, but to the best of some ENSO events. Now, in summary, I would like to say that 2 different approaches were proposed for predictability of ENSO events, and so that we can plan against its disastrous effects. And such results were compared with the dynamical prediction systems and the previous studies. And in conclusion, I would say that the proposed methods are exhibiting high skills in comparison, and that's throughout the long leads. And also they are able to cross the so-called spring predictability barrier. Also the skills for the different seasons are very much in variable range. They are not good for one season, not bad for one season. And also the changing climate dynamics is perhaps properly captured in one of the model, not the first one. And after studying the heat maps, you can see that the expected regions which are need to be contributing are observed to be contributing properly. Perhaps there is one limitation in the studies that the amplitude is still underestimated. We can see in this figure again that there is this 1997 event, the observed one is 2.5, but we're predicting only 0.5. So such underestimation in the amplitude is still observed. Perhaps we're working on this, and we are planning to improve that prediction amplitude also along with the direction. Perhaps the direction is already better, but it may be also improved by increasing the resolution of inputs. We have trained it on 5x5, but we are considering it on very high resolution. Then we're supposed to include more predictors and use a sophisticated algorithm for optimization algorithm for hyperparameters. These are references, and thank you for the kind attention. We can take questions, doubts, or comments, and I thank all my collaborators for their suggestions and inputs. Thank you.
Thank you very much, Dr. Kalpesh Patil. It is really wonderful talk. And if you have any questions, so kindly please write through the chat box. We have a time limitation. So please directly contact Dr. Patil for your questions. We'll move now to the next speaker. The speaker is Dr. Swan Naik from Manipal Institute of Technology. And title of her talk is Synthesis, Characterization, and Molecular Talking Studies on Isoxazolines. So, Dr. Naik, please start and share your presentation. So, is Dr. Swan Naik already there? Okay, if not, maybe we next move to the next speaker. Is Dr. Pawan Kumar Javaria, and from Beijing First University, and also he is now currently in Germany. And I'm pleased to invite him in the 12th ISAJ Symposium. And title of his talk is Molecular Mechanism of Apical Hook Development in the Arabidopsis thalia. So, Dr. Javaria, please start and share your screen presentation.
So, thank you, Dr. Santosh, for this invitation and introduction. Just a second, I make. So, are you able to see my screen? You can see, right, clearly?
Yes, clearly see your screen.
Okay.
Please.
So, okay. Then I start. Okay, I will talk about molecular mechanism of apical hook development in Arabidopsis thaliana. My name is Pawan Kumar Javaria, and I did this work in Professor Rishikesh Bhalerao lab in Umeå Plant Science Center, Sweden. And this work was supported by Beijing Forestry University, China. Currently, I am working in Professor Klaus Schäumer lab in Technical University of Munich. So in next 15 minutes, you are going to learn about importance of apical hook in dicots and what is the molecular mechanism of apical hook formation. It's not moving. Okay, so what is the purpose of apical hook formation? Apical hook is formed when seed germinate and young seedling emerge from the soil. It protects shoot apical meristem from being damaged. So why apical hook is formed and how it is formed? So it is formed by differential rate of cell elongation of hypocotyl. So you can see here that slower rate of cell elongation on the lower side and the faster rate of cell elongation on the outer side. So the hook development has three phases. The first phase is the apical hook formation. second is maintenance phase, and third one is the opening phase. So apical hook is formed in first 24 hours after the seed germination, when angle reached up to 180 degree. Then it is maintained up to the 72 hours. Then gradually it starts opening. The angle shown here in the blue color, you can see here, is the apical hook angle. In the next slide, I will show you a movie where you can see all these three phases. So after the seed germination, apical hook is formed, then it is maintained, then gradually it starts opening. You can focus here where you can see all these three phases. So hook is formed, this run is completely closed, then it is maintained up to the 72 hours, then it gradually starts opening. So this hook development is an excellent model to investigate mechanisms underlying differential growth. So hook development depends on carrier-mediated asymmetric oxygen distribution. Many of you know that DR5 is oxygen-responsive synthetic promoter, and oxygen response maxima is formed. You can see that GFP is accumulated only on the lower side of the hook, and this formation of this oxygen response maxima, oxygen transport, polar oxygen transport is necessary. Polar oxygen transport is maintained by oxygen influx and efflux carriers. OX and LEX are oxygen influx carrier, and pins are oxygen efflux carriers. And you can see in the mutant of pin 347, comparing to control, this apical hook is defective. And when oxygen transport is blocked by an inhibitor drug, NPA, then you can see there is no apical hook formation and also defective in the formation maintenance phase. So this suggests that hook development depend on carrier-mediated asymmetric oxygen distribution. So several labs have outlined the hormonal pathway involved in apical hook development, but nevertheless, what is the downstream target remains unclear. So as I explained in the earlier slides, that slower rate of cell elongation on the lower side and the faster rate of cell elongation on the outer side. So it is likely that differential rate of cell wall modification takes place. So we believe that downstream target is cell wall, and because of differential rate of cell wall modification, that differential rate of cell elongation may take place. To have this idea in our mind, we planned our project. So first I will start giving you a simple and probably simplistic view of cell wall. Cell wall is made up of cellulose, hemicellulose, and pectin. Cellulose and hemicellulose are cross-linked and they are embedded in pectin matrix. I focused mainly on MUR1 gene in my project. MUR1 encodes for GDP D-mannose 4,6-dehydrated. So MUR1 lack L-fucose in pectin RG2 and glycoglucan. So MUR1 plant were reported to be dwarf, and interestingly, addition of boron rescued them. Why boron rescued MUR1 this defect? Then it was suggested that boron stiffens the cell wall by making RG2 dimerization. And because of that, it rescued MUR1 defect. Then first we checked whether MUR1 has hook defect or not. Comparing to control, you can see that MUR1 has hook defect in the formation phase, and also it is defective in maintenance phase. Then we check whether this, because as I explained to you that MUR1 lack L-fucose in glycoglucan and pectin RG2. So this, whether this defect is due to RG2 or with glycoglucan. So there is another mutant available named as MUR2, which lack L-fucose only in glycoglucan. So we check MUR2 mutant also, and MUR2 is like wild type. This clearly suggests that MUR1 hook defect is due to RG2 pectin. Then next, we check whether exogenously added fucose and boron can rescue this defect or not. And we found because it is defective in fucose, when we added fucose, then it's comparing to MUR1, fucose is completely rescued. Interestingly, exogenously added boron also rescued MUR1 hook defect. Why boron rescued MUR1 defect, we completely don't know, but we believe that it also it stiffens the cell wall by making RG2 dimerization. So next question is whether does MUR1 hook defect due to failure to establish a polar oxygen transport during hook development mediated by PIN, OX, LAX, oxygen carriers. So first we check DR5. So DR5 is oxygen responsive synthetic promoter. As you can see here, comparing to control. So there is you cannot see much venous signal here comparing to control. So hook is defective. And very interestingly, when we provide boron, it goes up. You can see that in the MUR1, DR5 venous signal intensity is decreased, and in the presence of boron, it goes up. Then, because this oxygen response maxima, as I explained earlier, is maintained by oxygen influx and efflux carrier. So we checked oxygen influx and efflux carrier. OX and LAX, these two proteins are oxygen influx carrier. First, we checked the transcript level of OX and LAX3, and we found that OX1 transcript is reduced in MUR1, but LAX3 did not change. Then we checked the protein level of OX1, and we found that OX1 decreased in MUR1 too. And very interestingly, when we provide boron, it also goes up. Next, we check the transcript of oxygen efflux carrier. Pins are oxygen efflux carrier, and we found that pin 3, comparing to control, is decreased in MUR1, and pin 4 also decreased, but pin 1 and pin 7 did not change in MUR1 background. So selectively, pin 3 and 4 decrease in MUR1 background. So next, we check the protein abundance of pin 3 and 4 in MUR1, and we found that pin 3 comparing to control is decreased in MUR1, you can see, and in the presence of boron, it goes up. And similar to pin 3, pin 4 also decreased in MUR1 background, and when we provide boron, it goes up. So it is known that ARP7, oxygen response factor 7, and oxygen response factor 9 regulate OX and PIN's expression. So we checked ARP7 and ARP19 transcripts in MUR1 background, and we found that ARP7 and ARP19 both reduce in MUR1, and when we provide boron, it in both cases both goes up. Then we check the phenotype of ARP7 and ARP9, and we found that ARP7 and ARP19 also has defect in MUR1, like MUR1, and we made triple mutant MUR1, ARP7, ARP19, which show more severe phenotype comparing to MUR1. Next, we check whether if we provide boron, whether it can rescue or not. When we provide boron, then also the phenotype is similar to the triple mutant. So boron cannot rescue ARP7, ARP19. This data suggests that boron requires ARP7, ARP19 to suppress MUR1 hook defect. In 2020, our lab showed that there is when XXT, these two proteins regulate xyloglucane, and they also have defect in hook. And when they combined ARP2 with XXT, XXT2 in triple mutant, you can see that this hook defect is rescued. So ARP2 rescued XXT, XXT2 defect. So I also made a triple mutant, double mutant, sorry, MUR1 ARP2-8, and I found that it is rescued. But we don't know how ARP2 rescued MUR1 hook defect. So taking all this data, we can summarize in this way that MUR1 which lack fucose in the cell wall, and this defect is due to RG2 pectin. In this case, ARP7 and ARP19 goes down, and which probably regulate oxygen transporters pins 3 and 4 and OX1, and due to the decrease in oxygen transporters, oxygen response maxima is not formed in MUR1, 2, and which result in defective hook. And when we provide boron, probably it stiffens the cell wall, and then the ARP7, ARP19 goes up, and in this case, oxygen transport level also goes up, and which regulate oxygen response maxima, and proper hook is formed. I would like to acknowledge my my boss Rishikesh Balera and Beijing Forestry University for financial support, and thank you. Looking forward for your questions.
Thank you very much, Dr. Pawan Javaria. This is really wonderful talk on plant development biology, and it is good to see that you rescued a lot of plants by boron. So there is one simple question. I'm not a botany person, but how is specifically transcriptionally upregulating all the transcripts for this lower part of apical hook? It's not specific to the upper part where cell division is slower. It does make the symmetry in a folding structure. Is the boron dependent?
I don't know about basically about, but I can answer your question. So if you see this slide, so the outer side and inner side, then if you compare here, outer side the cell elongation and inner side cell elongation rate in wild type, okay? But in MUR1, 2, that outer side cell elongation rate is lower comparing to wild type. Here you can see, and inner side cell elongation rate is more in MUR1, 2. And when, of course, we combine with MUR1, 2 R2 dash, then it also like a wild type. So the change in cell elongation rate may affect this kind of transcription problem.
Thank you very much once again for your wonderful talk. And
okay, thank you.
If the audience get in touch by chat to Dr. Pawan Javaria, and we will move to our next speaker because of the time limitation.
Okay, thank you.
Thank you. Next speaker is Dr. Ullas Eskanam from the Jongsan National University, South Korea. And the title of his talk is Development of Novel DNA heptamer-based analytical tools to detect toxic insecticides from plants. So, Dr. Ullas Eskanam, please start and your presentation and share screen.
Okay. Okay, I hope you can see my screen, right?
Yes.
Yes. Okay. Good afternoon, everyone, and especially I would like to thank the ISAJ organizing committee for inviting me to give this talk. So today I'm going to talk about novel biosensors to detect insecticides from food and environment. And this work has been mostly conducted at the Gyeongsang National University in the Jinju. And here, if you are aware of the insecticides, you would probably know that most of the insecticides, they are ubiquitously applied in agriculture as well as the living spaces in the cities, in the houses, everywhere. Okay, so in past 3 decades, maybe like last 30 years, the use of these different insecticides or pesticides has been like increased by almost 200%, and this increase has caused several environmental pollution, health hazards, and especially the biodiversity loss. And because of this high consumption of insecticides or applications in the agriculture, we see increased food toxicity, and several long-term uses of this insecticide causes several health hazards like diseases like cancer or autoimmune diseases, as well as memory loss. So the problem with this using of different detection methods that available today in insecticide analysis is these traditional methods, which are like HPLC, GCMS, LCMS, and MSMS. So all of these methods are very good. That they have very good sensitivity, or they are very accurate. But the problem with these methods is they are very fixed. They are immobile. They cannot move from one point to other point. These are they require a heavy instrumentation. Most of these methods are very expensive, costly. You need millions of dollars to buy HPLC instrument. The procedures, sample preparation and analysis is a very laborious process. It is time consuming, and sometimes based on non-traditional samples, the accuracy or sensitivity suffers. So this same method cannot be applied for the all kind of samples, and most of times you need a skilled technician to do the analysis as well as perform the experiments. So we are developing novel aptamer based biosensors which meet the assured criteria. So what is the assured criteria? That means the technology for the detection of insecticide toxic molecules, which are very small molecules. That technology should be very affordable in the sense of time as well as money, so you can easily do the analysis. The method should be sensitive as a part of assured criteria. It should be very specific, so that it should be able to discern or distinguish between similarly looking structural molecules. Okay, and then this method should have a rapid, quick, and simple analysis, and it should be robust, so that you can analyze any type of sample like plant tissue, soil, water, or food. And it should be easy for the end user, like a farmer or a small or a chef in a restaurant should be able to use. And the most important point of this assured technology is that it should be the equipment free. So you can take if we develop this assay into LFA based, like a pregnancy kit or something, so it should any end user at a point of care, it can be used as a point of care tool or technology. So these biosensors, which are aptamer based, they bring many advantages over traditional methods. But the key factor that is limiting in usage of these aptamers is how to identify an appropriate aptamer. And this process, or this question, or this problem was solved by Turk and Gold in 1990. And they published a very nice paper in Science, and based on that they also further developed many patents and also formed company. So the SELEX process, which is like a systemic evaluation of ligands by exponential enrichment, that is the basis for the aptamer discovery. And it depends on single-stranded DNA library, which can be in vitro synthesized, and this library contains of like 10⁻16 molecules, and this library is attached to streptavidin binding resin, and once this binding is performed, then we introduce a different chemicals, target molecule and non-target molecules for the selection, and like 15 to 18 rounds of selections are performed, and this 15 to 18 different rounds, they give a very highly specific single-stranded DNA which recognizes target insecticide or target molecule. And the advantage of this methodology is it can be optimized for any toxic or even small molecules. For example, if we use the toxic molecules in case of antibody raising or peptides, that cannot be done because of the toxicity. Like most of the times for generation of antibody, we have to use animal models or some live models, and in that live models or animal models, toxic compounds cannot be used. So that, but in case of selects, you can use any toxic molecules or any small molecules. There is no limitation. The only limitation is you have to just perform higher number of selections, and based on the selections you can find out the aptamers. So once the aptamers is find out or selected, then we do the bioinformatics analysis, and the bioinformatics analysis is performed to find out the 3D folding as well as the free energy change, so that which aptamer has the highest stable structure at the ambient conditions like 25 to 30 degrees Celsius. And based on this, then we design fluorescent probes. So the fluorescent probe design consists of attachment of fluorophore, like in this case it could be 6 FAM, that's 6 fluorescein, and it's quenching using a quencher molecule like a dapsil. And then once this is synthesized, this this quencher is allowed to bind to this aptamer, and once the quencher binds and to the aptamer, it forms a aptamer quencher complex, and in presence of quencher, the aptamer, which is fluorescently labeled, it doesn't produce any signal. Okay, but if the target molecule, so insecticide, in this case fipronil or any other insecticide is present, then this aptamer can bind to this insecticide and the quencher is released. And once the quencher is released, you can get the signal, and that signal can be quantified and used for the detection of contamination of food or soil or water, any environmental factor. And then we can say whether particular insecticide or pesticide or any toxic chemical is present in the food or not. So based on this aptamers, we also showed that these aptamers can detect presence of these chemicals in food samples. And for this we used several vegetable samples as well as fruit samples, and we calculated the KD, that is the binding affinity. So for this purpose, we perform the KD analysis in 2 steps. So the first step is the KD KD1 or KD effective 1. So that KD is calculated based on the quenching efficiency. So once aptamer, the fluorescence tagged aptamer binds to quencher, that can be represented as a KD1, and in presence of fipronil, the quencher is released, and that can produce a second KD, that is a KD2. And then we finally calculate the final KD, that is a KD1 over KD2, and this will give us the ultimate or the final KD value. And we found that these KD values are very sensitive in a sub picomole or sub ppb level, and these are at the industry level sample. The another aspect of any novel technique or novel biosensors is it should be able to recognize the specific sample with a very high specificity. And in this case, we used non-target molecules or structurally similar looking other chemical compounds, and we found that our sensor can distinguish between the specific fipronil and structurally looking its similar compounds with a very high specificity. So this proves that our sensor is very specific. Additionally, we developed a label-free sensor for the fipronil detection from various plant tissue extracts. And for this, we tested the ability of our aptamer to form a special structure called G4 quadruplex structure. And based on this G4 quadruplex structure, if the aptamer forms this structure, it can react with a THT dye. And this based on this reaction, it will produce a signal, fluorescent signal. If it doesn't form a structure, then it cannot form. But luckily, our aptamer could form this quadruplex structure. And when you add fipronil to this structure where THT is already attached or bound, since fipronil has a higher affinity, it can easily replace the THT and the signal is lost. And this way, the presence of fipronil, which is a toxic compound in the solution or in the food or in environment or any wastewater, we can detect this presence of this chemical or any chemical, and then that signal can be measured. And this measurement of signal is very concentration dependent, and it can be quantified. So this label free assay is also possible using the aptamers. Then what we did is we wanted to find out what is the specific binding pocket in this aptamer. For this, we performed site-directed mutagenesis of guanine residues and some other residues, and for this we used molecular modeling. Okay, and based on this molecular modeling, we found out that we used three different molecules: THT, fipronil, fipronil sulfate, and we found out that our aptamer had highest affinity for the fipronil, and then THT, and then lowest to the non-target molecules. So this is a very good study which shows that particular aptamer can highly, with high specificity and high sensitivity, can detect the insecticides. Then again, we performed a biophysical analysis to check the 3D folding or the conformational changes in the aptamer based on how it interacts with the target molecules and the non-target molecules. And then we found that yes, the reactions of the FIP-A6B aptamer and the mutants are highly specific, and they are highly specific in response to the particular target chemical. And here I'm showing only representative images of FIP-A6B with THT, fipronil, and combination of THT and fipronil. Then another aspect of developing a novel sensor or novel tool is that you must validate this tool with existing gold standard method. And the current gold standard, industrial gold standard method for pesticide or insecticide or any small molecule toxic compound analysis is the HPLC. So we validated our experiments with HPLC, and we found that our method performed much better than the HPLC analysis. Here you can see that in HPLC on the left side, the HPLC graph is shown where the LOD, that is limit of detection and limit of quantification for HPLC for fipronil from the plant tissue extract is 8 nanomole and 25 nanomole. Whereas during using our assay, we show that the LOD is almost half of the HPLC, and same is true for the LOQ. So this proves that our assay, our novel biosensor assay or DNA-based apta sensor is highly specific and highly sensitive against the specific compound, that is a fipronil. And moreover, we showed that our assay, this biosensor, is 100 to 200 times cheaper than the commercial HPLC method, and it can also perform much faster. For example, in case of HPLC, you can do only one sample at a time, whereas in case of our assay in fluorometer, we can do 96 well plate or 394 plate at a time. So that way you can save a lot of time.
You may please conclude. Sorry.
Okay. With this, I would like to bring it to the final slide, that is a summary and conclusion. So here I would like to briefly conclude that we have developed a very sensitive and specific assay which can detect fipronil from plant tissue extract. We developed two different modes of detection: fluorescence labeling and label-free. This assay achieved very high sensitivity, and they performed better. Finally, I would like to acknowledge my professor for giving me this opportunity, and different funding agencies, and my colleagues, especially Kian from Vietnam, and other lab members for supporting or providing me help during this work. Here, I would like to conclude, and I would like to open the floor for the questions.
Thank you very much, Dr. Vlasis, for sharing your interesting findings. And this session can have one quick question from audience. Or please directly send questions to Dr. Kata by chat.
Okay, I have to exit. No, how can I?
Thank you once again, Dr. Vlasis. And we have next speaker, Dr. Chimli Bhattacharya from NIT Nagaland. And the title of her talk is on elucidation of structure-function relationship of human proteins with the sulfur drugs. So Dr. Chimli Bhattacharya will share screen and start the presentation. We can see your screen, so can you start speaking and start the presentation?
Good afternoon to everyone. Thank you, Dr. Shantos, for introducing me, and I'm grateful to. Sir, my screen is visible, right? The whole screen is visible.
Visible. Can you please make it presentation mode, full view of your slide?
Yeah, I have already done it.
Okay, so please go ahead.
Is it okay? Is it visible?
Yes, it is visible. So please start.
Okay. Thank you. Thanks to the host once again for his introduction and kind help. And I would like to thank Dr. Kedarnath Mahapatra sir. I'm connected, and the entire ISAJ team. I am connected to ISAJ for last more than 12 years when I was a postdoc in Konan University, Japan. So it's always a very good feeling when I can again attach with ISAJ and I can present. I am thankful to the team for again giving me an opportunity to present my own research. Today, I'm going to talk about the elucidation of structure-function relationship for the of human proteins upon binding with sulfur drugs. Presently, I am working in National Institute of Technology, Nagaland. It is in the far east of India, which that region is called at Northeast. There are 7 states. Among that, we are in the far east position. My work is this work is joint with my scholars, Mr. Aben and Miss Mavani, and we have also done a few portion of work in IICB CSIR Institute of Chemical Biology, Indian Institute of Chemical Biology in Kolkata. So this is just a small glimpse of the place where I stay now. We are into a green paradise. It's a very nice place, excellent weather, excellent scenic beauty. Our institute is also now in a developing stage. So we first started to focus on certain problems or research problems which are associated with Nagaland or the northeastern India. Obviously, these places are a little bit quite underdeveloped compared to the mainland India. And as I am basically from Calcutta, born and brought up till PhD, I was in Calcutta. Then I moved to Japan for postdoctoral studies. After coming back, I joined here as an assistant professor. I can clearly see the difference. Though there are huge resources, the major problem is the lack of awareness. What I felt, that is, there is a lack of awareness. Like there is a huge antibiotic pollution. The pollution problem is here. And among the different kind of pollutions, the antibiotic pollution is going to be, and the funniest or the most interesting part of this area is the people, the place, the administration, none of them are very conscious about the pollution problems, about the sustainability of environment. As it is very rich in biodiversity, it is considered to be one of the hotspot of biodiversity place, center of hotspot of biodiversity of India. So huge resources, natural resources are there. So people are not very concerned about the problems which are happening here. That is going to hit the state as well as the entire region, maybe after 10 to 15 years. By that time, it will be too late. So we started working with few antibiotics. Like these days, antibiotics are coming into the category of excuse me, please. I'm not able to move my screen. So my screen is not moving. Is it like that?
Yeah, actually, to me, your screen is not moving. I don't know for all members.
Me also, it's not moving. What happened? Sir, please excuse me. Maybe my network is not very good.
You choose slides by your mouse.
Yeah, my slides are not moving for some reason. Yeah, just a moment.
Yes, we can see moving.
Yeah. Now it's okay. So we have to look upon the antibiotic problem, which are the one of the most major emerging pollutants. Not only antibiotic, different kind of drugs. We see that the drug is expired, or for some reason we just throw it, we just expose it. And here the disposal system is not very strong, so that we are throwing everything in a single place. So we have checked it. We are mainly working from two aspects. One is the environmental aspect and one is the biophysical aspect. So in the environmental aspect, we have already tested all the wastewaters and all. We have seen a huge quantity of different kind of antibiotics present in the system. Among that, one is sulfanilamide group. Sulfa group is a very common antibiotic. Sulfa group is very common and vastly used. And Nagaland or all these kind of states, they are mainly into the they rear different kind of animals like pigs and all these farms are there. So they use this kind of antibiotics in a vast manner. And after that, I think they are directly going to the environment. And in many cases, in the fleshes of the animals or in the milk of the animal, this kind of antibiotics are found. That is from literature survey. We have not done that part in the real life. So what we try, how this kind of antibiotics, when they are going into the environment, and if it is coming to the human body through some food or something like that, in form of food, water, whatever, how it is going to affect the thing. We know that antibiotics are mainly, they are organic molecules. Being organic molecule, they are planar in nature. Benzene rings are there. These are excellent intercalators or groove binder. They can bind with any kind of biomacromolecule, either DNA or protein. So we have here focused on the protein. We tried to see how this kind of antibiotics are binding with protein and how they're affecting the structure. Finally, how the structure and function are both affected. So we have mainly worked with hemoglobin, myoglobin, human serum albumin, Brovin serum albumin, and lysozyme. So usually we started with I will just show one set of data of one set of protein, and I will go for a in general discussion. So I'm showing the hemoglobin one. Hemoglobin, we know it is the most major important heme protein of our body. It is the transport protein. It is actually transporting oxygen into different parts of the body. So as myoglobin, it is also another heme transport protein. So we tried to check, and these are the serum albumins. Separately, I have shown once again. The properties are mentioned. I'm not going to discuss those about this. Everyone, it's known. And this is the lysozyme one, and this is the drug. This is basically the drug moiety, sulfur drug, which we call them. They are nicely binding with the protein, and we have started with the UV visible spectroscopy, and we have shown that there is a significant perturbation. We have mainly taken two kinds of sulfanilamide drugs. One is sulfamethoxine, another is sulfadiazine. They are almost same, but little bit different into the methyl substitution. So we have also tried to check how this kind of substitution in the drugs are affecting the protein structure. So obviously from this kind of work, what the major thing is coming, we generally say that, yeah, it is harmful. It is binding. It can affect the function, but how the quantitative part, we have tried to focus on this quantitative part. And for that, as I have told, that we have started with the UV visible spectroscopy. We have seen there is a significant perturbation in the structure. We have calculated the binding constant. The binding constant comes into the order of 10 to the power 4. It is quite high. With respect to protein binding, 10 to the power 4 is moderate to, it comes in if the binding constant, the equilibrium constant, we have calculated from the UV visible spectroscopy using the Bensey-Hildeberg plot, and that is a technique of analysis we are using, a mathematical model. And for that, we can clearly see that there is a significant amount of perturbation in the structure. And as I have already mentioned, for protein, 10 to the power 4 order binding is quite moderate. It comes into the moderate to high binding only. This is the general most common binding order. Finally, we are going to correlate with the oxygen binding here. Then we have checked the thermal stability of the protein, again with UV visible spectroscopy only. Well, surprisingly, we have seen that compared to free protein to the bound protein, bound with the drug, the TM, the melting temperature, is increasing from 61 to 65. It is very high. That means more than 6 degree change in the melting temperature. So once this, that means there is a huge stabilization in the protein structure upon drug binding. That in that case, what will happen when oxygen will come? Ideally, the hemoglobin proteins work is to bind with oxygen. Binding oxygen capacity is actually much less than this kind of drug binding. So when the drug becomes, when the protein becomes thermally stable after this kind of binding, of course, it will not be likely to release the drug when his actual partner. Why I'm saying actual partner? Because we always see that it is the work of the hemoglobin to transport oxygen. Oxygen will not get a place. So we have tried with synchronous fluorescence. We have checked with the fluorescence. From the fluorescence also, we have calculated the apparent binding constant. We have done the Stern-Volmer analysis. Then we have done FRET. In FRET also, we have shown that in FRET, the receptor and the donor, means the protein and drug, the binding is actually, the binding distance is around, the distance between the donor and receptor is 3.75. That means usually if it is less than 8 nanometer, we say that, okay, this is a very high scope of binding. And here we can see the R value is much, much less than 8 nanometer. That means they are significantly interacting with each other. We have done synchronous fluorescence also, and then we have done CD analysis, circular dichroism. Everywhere we have shown that the alpha helix portion of the protein has been affected significantly. So finally, we have done the IR spectroscopy. These are for the structural part. The synchronous fluorescence, the IR, all the CD kind of data. These are for the structural confirmation. And then we have again for the functional part, we have moved to calorimetry, isothermal calorimetry. There also we are getting the binding constant in the order of 10 to the power 4. Finally, we have done the molecular docking studies. In molecular docking study, we have seen that where these are actually, where the tentative place, tentative position, where it is going and getting attached with the protein structure. And from that, it is again clear that it is going to the very near to the heme group where the main oxygen binding is happening. So we have already reported this data in Wiley can select the paper already last year 2020 only it has been accepted, and we are now moving for the next drugs, next proteins, which I'm going to show that 1:1 stoichiometry, more or less the results are quite comparable. Generally 10 to the power 4 order of binding, 1:1 binding stoichiometry, significant change in the conformation, significant change, quite significant moderate to high binding constant, binding affinity. So here we have shown that it can disturb through just a scheme. We have this part is under progress. We are going to do this experiment. We want to do this competition essay to see that if really what is happening, if oxygen is also present in the system and this kind of antibiotic drugs are present in the system, how the hemoglobin protein will react. So we are going to check that part as well. So that is under progress, but this is the scheme where we have already predicted that how it can affect the binding of the oxygen, binding of oxygen and hemoglobin binding. Then we have done with myoglobin. I am not going to show this data, only one part I will just show that in case of myoglobin, we have done with two drugs, sulfametazine, sulfadiazine, and we have shown the homolumo difference when we are going for DFT studies. The homolumo difference is within the order of, so it is in the order where there is pi to pi star electron transition is quite obvious. And of course, it is also another support that this kind of drugs can bind to the protein very successfully. So these are the, yes, ma'am, sir, I'm almost done. Only 2-3 slides. So like that, we have tried with the hemoglobin, myoglobin, then we have tried with human serum albumin, as well as we have tried with the lysozyme as well. So both the drugs we have tried, and some other drugs are also under progress, under investigation. So from here, we can just conclude that we should be we are giving a picture of structure function, how the structure is being affected, and finally the function can be affected for this kind of very important proteins of our body. So we want to give the message that we should be careful about this kind of antibiotic overuse. Moreover, their disposal procedure, so that we will not be landing to a much difficult situation. That's all. This is the papers we have already published, and some are under communicated stage. So again, it is the end. Here I am just ending my presentation. I would again like to acknowledge Dr. Kedarnath Mahapatra sir and the entire the ISAJ team, Dr. Santosh sir for all the hosting and everything, and our collaborators, Dr. G. Suresh Kumar sir, Professor Daisuke Miyoshi from Konan University, Japan, and of course the funding agencies. First of all, our institute director, NIT Nagaland, and our one grant we used to get from TechIP 3, and of course the other funding agencies like BARC, DBT, DRDO. Fortunately, we are getting good research grants. So thank you once again to everyone who have supported us for doing this work. Thank you.
Dr. Bhattacharya for a wonderful talk and letting us know on the properties of those molecules on the DNA. So thank you very much. And if there are questions from audience, please write chat directly to Dr. Bhattacharya. Due to the time limit, we'll move to the next speaker of our session. Was Dr. Dipya Pramanik from Gyeongsang National University in Korea. And the title of his talk is CRISPR-Cas9 mediated genome engineering of tomato for pathogen resistance trait. This is a case study. So, Dr. Pramanik, please start your presentation. And you have 15 minutes, including question and answer.
Yeah. Good afternoon, sir. Am I audible?
Yes, you are audible, and we can see you.
I hope. Okay. Okay. Thank you, Dr. Santosh, for my nice introduction. So, and first of all, I want to thank the organizing committee of ISAJ for giving me the opportunity to present my work in this symposium. So, my name is Dipjyoti Pramanik, and I'm a postdoctoral researcher at Professor Jane Kim's lab in Gyeongsang National University, South Korea. And today I will talk about the CRISPR-Cas9 mediated genome engineering of tomato for pathogen resistance trait, a case study. So plants are actually incisal in nature, so that means they cannot move. So they like us, plants are also surrounded by many microbes like majorly bacteria, fungus, or algae, and they are sometimes they enhance or promote the plant growth. That those are called beneficial microbes, whereas a part of microbes that can significantly infect and produce a disease that reduce the yield. So those are called harmful microbes. So why we are talking about this plant microbe interaction and why we need to study more about this plant microbe interactions, and including to develop a more better crop for future. So actually, when the pathogens attack the plants, it not only infect but also it reduce the yield. So think about in the global situation for crop yield, it reduced from 30 to 100%. And if you see the recent report of World Report, maybe by 2050, we will have increase of 10 billion people, and we need 56% more food than current time. So that means there is a serious attention is required to not only develop a new varieties, but also the ways to tackle the situation and improve the overall production rate. So one aspect is if we can develop a pathogen resistant varieties with a quick time, that can be one solving things. So today I will talk more about tomato. So tomato is one of the important vegetable crop and also a model system for plant biology. So apart from, as I said before, tomato is also surrounded by many pathogens like viral, fungal, or even bacterial. So today's talk mostly cover with two diseases. One is caused by one viral pathogen that is called tomato yellow leaf curl virus, and other one is powdery mildew disease that is caused by one fungus, that's Odium species. So first one is the TYLCV. TYLCV is actually, people called it as a devastating virus, those who are working with tomato. So as you can see in the figure, these are the common symptoms of TYLCV, of curling and yellowing of the leaf stem that reduce the yield. So this field trial, as you can see, the resistant varieties, which is developed by using crossing, that takes actually several years, and subtotal that variety that cannot tackle the infection and reduce the yield here as a 30%. So in recent years, it is reported that the TYLCV infection is spread throughout the world. So this is the current map. You can see where the TYLCV infection is already spreaded, and that includes the major tomato producer nations like India, China, and Korea. So TYLCV infection is spread mostly by white flies. As you can see here, these white flies, when they consume the nutrient from plant, it transmit the TYLCV viral particle to the plant host, and after going inside the plant cell, it will hijack the host machinery and gradually proliferate and multiply and transport from one part to another part, and the disease can be formed. So in a couple of decades, there are several reports we found that showing the resistant traits that means one locus or one gene that can tackle this TYLCV infection. And out of TY1 to TY6, we found that TY5 locus that carrying pelo gene is actually a messenger RNA surveillance factor called as pelota. It plays an important role in viral replication. And when it is downregulated in wild tomato cultivars, it is observed that plants are resistant against this viral infection, whereas overexpression increases the pathogenicity of the virus infection, as you can see here in the curling of and yellowing of the leaf is visible in the upregulated below mutant plants. So another disease I'm talking about is powdery mildew disease. So as maybe you can already know, this kind of powder-like structures in the leaf surface or in the stem, these are actually of fungal spores. It's also infect major crops like barley, wheat, soybean, and many vegetables crops and fruit crops, including and tomatoes. So the powdery mildew resistant locus is already well characterized. So as you can see here, this is actually a transmembrane protein that helps to progress the viral fungal infection in the infected plants. So already we found there are a couple of reports that showing the targeting this MLO gene or downregulation of this gene can produce a resistant phenotype. So for the trait integration purpose, we also target this one. So there are several techniques available to make a knockout or deletion of one gene function. But why we are using CRISPR? Because this is a recent, I think 2012 around, it's published, and later it is well adapted for several applications. So this CRISPR system is actually a native integral part of bacteria or archaea that use when any bacteriophage infect, and to protect the bacterial system, the system is activated. And later, the system is well adapted, and several tools were generated that can be targeted, that can be used to target or modify any target genome, including animal or even plant. So there are several reports we already know that showing the application of CRISPR tools for several applications. So here, some of the recent reports where the CRISPR generated or edited plants can be already commercialized, like US FDA approved non-browning tomatoes, non-browning mushrooms, and also the high GABA content, the tomatoes. This is actually, this high GABA tomato is already, I think you must be heard of, it is already commercialized in Japan. So for among all the CRISPR-based tools, we use the most common, the CRISPR-Cas9 system. This is a simple two-component system carrying the Cas9 nucleases, and another second part is the RNA, which is specifically we can design to target any genomic target of any cell. So initially, we designed a couple of guide RNAs that can target the coding part of or exonic part of the cell payload gene, but we could not get any success. Later, we designed several guide RNAs to target, and among all 9 guide RNAs, we found one guide RNA work efficiently, and which have made genome-edited lines. Similarly, we designed a couple of guide RNAs to target SLMLO1, and these are the CRISPR-Cas9 vector constructs we made, and we transform into agrobacterium, and we deliver this agrobacterium to transfer this CRISPR component carrying plasmids to the plants. And we generated by using tissue culture, we made transgenic or genome-edited lines, and we for the TYLCB pathogenicity testing. Actually, as I told you before, that the TYLCB carried by the whiteflies, but in the lab we want to test this by mimicking the similar situation. So we used one reported construct that carrying the viral particles, and by using injection we infect the plants, and we observe the phenotype. So after 28 days of post-infection, we observed very severe level of infection rates in wild-type plants. So later we extended our work to evaluate or test our plants. So here I tested 3 lines that carrying 1 base addition. As you can see here, this is the wild type DNA, and the guide RNA is in the red color, and only 1 base addition that cause a frame shift mutation and a premature stop codon, and we can delete this gene function. So when we check the phenotype after 28 days, we observe there is a reduced infection rates. And when we check the RT-PCR by designing specific primers to target the viral component, we observe there is a significant reduction of the viral load in comparison to wild type. As you can see here, wild type is severely infected, whereas all the genome-edited lines are quite resistant against this viral infection. So similarly, the tissue culture generated plant we made, and then we segregate in the next generation and characterize for powdery mildew. And here we check also with ODM species, and we observe that wild type having severe infection with powdery mildew, as you can see here in this picture. And similarly, for 3 lines we tested, it shown complete resistant against powdery mildew. And further, we also checked the ribosomal sequencing to confirm this viral infection. And we also wanted to be safe, we also wanted to check any off-target effects. And we checked several off-target sites of all the guide RNAs, but we could not detect any off-target. So that's a good point. And later we also wanted to get some tDNA-free line. So we also characterize, like here, 1 line that don't have tDNA insertion. And this mutant line is having larger deletions where there is no any foreign DNA present in the genome. So in summary, we established the CRISPR-Cas system to target 2 susceptibility genes. One is SL-PELO and one is SL-MLO1. And we also observed the mutant plants are, or genome-edited plants are resistant to respective pathogens. Like one is for virus, one is for fungal. And in a next thing we actually seriously need to do, that is a field trial, because till now, whatever test we did in the greenhouse or even the lab condition. So we have to check those plants and their resistance ability in the field. So another issue is the GMO, and the regulation issues. So till now, maybe there is not a clear regulation guideline for the Korea. So maybe we will hope to get clearance of these genome-edited plants for further commercialization. So recently we published this work, and at the end, I wanted to thank my PhD supervisor, Professor Jane Kim, and my fellow lab mates for those who are involved in this project, and the collaborator, and of course the funding agencies for giving the fund for this work. And with this note, thank you for your attention, and I welcome any questions.
Thank you very much, Dr. Pramanik, sharing your research on CRISPR-Cas9-induced gene editing and targeting the susceptibility genes of tomato causing various diseases. So this session is open for questions now. We have 2 minutes, so if audience have any question, please ask. So you can directly ask your question through chat, Dr. Pramanik. And thank you very much once again. This is very interesting talk on genome editing of tomato. And for this, we are almost ending this session for me. And I want to again recall if there is Dr. Sangori Nayak from Manipal Institute of Technology, if present in this session, please share your screen or start presentation. If present here. So I guess she is not available in this meeting. So I think that's all from our side for the session 4B. And thank you once again for all the participants. And we will soon see you in the session 5B, which will be starting from 4 o'clock, 4 p.m. So after 15 minutes, we will resume the sessions. Thank you very much for your patience and active participations. See you at 4. Thank you.
ISAJ Annual Symposium. We will start very soon. Our first speaker, Mr. Vinay Shimpi from National Institute of Technology, Puducherry, India, will be presenting response surface-based model updating of heritage multiple art gallery, Masnili Bridge. So please, Mr. Vinay Shimpi, you can start your presentation with full screen.
Good afternoon, everyone. Am I audible to you?
You are audible and you can start video as well.
My slides are visible? Hello?
You can start now, and this session is for 15 minutes. So,
Okay, so hello everyone. Myself Vinay Shimpi. I am a PhD research scholar in Department of Civil Engineering, National Institute of Technology, Puducherry, India. I am presenting here on the topic of response surface-based finite element model updating of heritage multiple arch gallery masonry bridge. So, for our study, we have taken a bridge on the Kalka Shimla mountain railway. If I tell you about the Kalka Shimla railways, the Kalka Shimla railway is more than 125-year old, and it is in 2000 declared as a World Heritage Site under the Mountain Railways of India. The Kalka Shimla railway is 96.6 km long and a single track railway line. Here, the total more than 800 arch bridges and more than 102 tunnels are there, which are still in operational. Out of which bridge number 541 is the tallest multiple arch gallery bridge available here. So our study will be concentrated on this bridge. This is the bridge picture. As you can see, this is not a conventional arch bridge. It is a multiple arch gallery, and we had done some primary investigation on the bridge, which says that the bridge was built in year of 1895 to 1900, and it got operational from 1905. In 1905, this bridge also experienced the earthquake, Kangra earthquake, which was very devastating, but it survived. Recently, in 2014, also this bridge survived with the Nepal earthquake. So since this bridge is located in seismically active area, the assessment of this bridge was necessary, and we did some primary investigation, geometric investigation on the bridge. So the bridge number 541 on Kalka Shimla railway is located at Kano, which is approximately 1833 meter above sea level, 1833 meter above the sea level. The bridge is having total 13 arches at the top floor, that is stage 1. Then it is having 9 arches stage 3, then 4 arches at the stage 2 and 4 arches at stage 1. The total length of the bridge is approximately 52 meter, and each arch is having a span of 3 meter, and the plan of the bridge is on the reverse curve of 48 degree. The bridge is constructed on the hard rock strata, and from the visual survey of the bridge, there were no sign of any damage or crack or dislocation of elements were found. Hence, it was assumed that the bridge is in undamaged condition. Now, the experimental campaign. For the experimentation, we adopted ambient vibration testing. In recent years, AVT has become most common adopted field measurement technique, which consists of recording the response of the selected structure under ambient noise like wind, traffic, or operational condition. Generally, the testing is performed using sensors like accelerometer, velocimeter, seismometers, and the response of the whole structure is recorded. For our bridge, the dynamic parameters are recorded through uniaxial accelerometer, which is shown here. This is the we used Magit and Decomet, and then 6-channel data acquisition system we utilized to save the data. Then the placement of the accelerometer over the bridge was selected based on the modal analysis of our initial numerical model. The configuration of the accelerometer was set up in such a way that the translation and vertical modes of the bridge can be estimated. Based on the numerical analysis, total 35 locations on the bridge deck were selected in vertical and transverse direction to take the readings. Here you can see the red position. Here 2 accelerometer in transverse and vertical directions were set as a reference accelerometer, and the 2 were moved from 1 to 35 to cover the response of the whole bridge. And here you can see our team placing the sensors with the high powerful adhesives. Here the placements of the sensor in the data acquisition system taking reading. The reading were taken when due to the train induced vibrations only. So for the bridge, total 70 minutes reading were required and processed for the next thing. And now for the estimating the modal parameters like natural frequency and the mode shape of the structure, we utilized operational modal analysis. Actually, operational modal analysis is a numerical procedure which is used for the processing the collected acquired data from AVT, which covers the entire frequency of the modal characteristic of the structure. The objective of the operational modal analysis is to extract the resonance frequency damping of the structure. This method is sometimes called output-only modal analysis. Here we utilized, if you can see here, the output response PSD will be available, and we will, with the natural excitation, we will create a frequency response function. After creating frequency response function, we will decompose into singular values so that the peaks will appear in the singular value diagram, that is modal identification function. The peaks will show the modal frequency, and the vector of that peak will show the mode shapes. So based on this philosophy, we used frequency domain FSDD frequency spatial domain decomposition method to estimate our mode shape from the acquired data of AVT. So this is the identification plot for the bridge number 541, where we can see the first, and these are the mode shape we got. Actually, we estimated 3 mode shape from our reading. The first mode shape is the bending mode shape at the frequency of 4.65 Hz. Second is the second bending mode shape here, which we got at the frequency of 11.38 Hz. And third is the vertical mode shape we got at the 23.85 Hz. Next, numerical modeling. The initial 3D model of the selected bridge was developed in Abacus. The geometry of the model was defined adopting the structural drawings available with Shimla Division Northern Railway, and further visual inspection and geometrical investigation carried out earlier to measure some elements. After the investigation, we found that the bridge contains 3 structural parts. One is pier, then top wall, and the arches. So these 3 components we modeled in our numerical Abacus environment. The piers were modeled using C3D8R element, which is a hexagonal element, as you can see, because the masonry in the arches and the piers were consistent and regular. Though the masonry in the walls were irregular, and due to its complexity shape, we used hexagonal element. Further, all the elements we created and we joined them with a tie constraint so that it will give a rigid connection response. As in visual inspection, we found there is no delamination or joint connection damage in the bridge. Further, to act for the connection of the bridge with abutment was rigid, so we provided fixed boundary condition at abutment, and also at the foundation also there was no displacement found, so no settlement found. So we provided a fixed boundary conditions at the bottom of the piers as well as in the side for the rigid connection Further, in this study, for the material modeling, we used macro modeling strategy where the unit of the masonry and the binding material were homogenized into the equivalent continuum. This approach is more practical for the analysis of the large heritage structures with the complex geometry. And also this methodology reduced the computational cost. These both bridges were constructed with the limestone, with the lime mortar. With the limestone used were the dressed regular stone for piers and irregular stones for the wall. The material property is also shown. For the limestone masonry, we used 3 parameters: Young's modulus, density, and Poisson's ratio. The Young's modulus we choose here is 10,000 MPa, density 2400 kg per meter cube, and Poisson's ratio 0.2. After developing model, the dynamic characteristic of bridge we estimated through the modal analysis, and here we compared the results of our initial finite element model with the experimental results. If we see, the mode shape of the both are same, but the natural frequency where they are got are different. Like for the initial model, we got the first bending mode frequency at 4.13 Hz, but for the experimental one, we got 4.63. So there we got the difference. So we can say that our developed initial model is not reliable. There is a necessity to calibrate it. So we started our model updating, finite element model updating. So what is finite element model updating? Finite element model updating is usually the procedure to reduce the results from experimental to numerical model so that we can get a reliable numerical model. The cause of this error may be the model structure construction, their model parameters, the measurements, or the model order or material properties. The selection of parameter for the modifying is a very crucial part, because if we change any other parameter, it will not give a realistic model for our structure. Generally, the material properties like Young's modulus and material density and Poisson's ratio can be selected and said to be very uncertain parameter in case of heritage structures. So for the finite element model updating, we used response surface methodology. The response surface methodology is a statistical procedure which estimates the response to the input parameters. For the FE model updating technique, the uncertain parameters are taken as the input parameters, and the error parameter between FE model and experimental results are taken as the response parameters. So here is the flow chart how the response surface method for finite element model updating works. First, we will select the features which are the input parameters and which are the output response. Then we will screen the parameters based on their sensitivity to our input response. The sensitivity of input parameters to the output response will be checked. Then a RS mathematical expression or response surface model is created, and we will check the adequacy of the RS model. If the response surface model is adequate, then we will go for the some optimization technique to update our finite element model and modify our input parameters so that the discrepancy between the numerical results and experimental results can be removed. So for the feature selection, we have selected 6 parameters based on the material properties like Young's modulus of stone masonries for piers, for arches, and for walls. And then sensitivity of the selected parameters on the response, the efficiency of RS model in which the output response are sufficiently sensitive to the input parameters. The insignificant input parameter should not be considered for the construction of RS model. In our study, we use sensitivity analysis. Here is the formula we use. Xj is the model parameter, Ri is the response of that output, and del Ri is the range, and del Xj is the range of the input parameters. And we got result for our 6 variables, where we can see the most influential parameters are Young's modulus of the piers and density of the piers, whereas the very less influential parameters are Young's modulus of wall and density of a wall. But still they are significant because they are 10% sensitive to our response, like modal frequency of our structure. Now after the selecting our input parameters, we have to design of experiment. So for in this study, we used central composite design, which contains factorial point, center point, and axial points. A total of 90 runs, which considered 64 cube points, 12 axial points, and at center point in the cube, and 6 center points in the axial are considered. The whole bridge was divided into 3 parts: pier, arches, and wall, and different material properties selected and adopted as the input parameter. The output response was selected as the 3 model frequency, which we estimated from experimental results. Therefore, Y1 here is the first bending mode shape frequency, Y2 here is the second bending mode shape frequency, Y3 here is the first vertical mode shape frequency. And we did numerical in Abacus, we changed the parameter, got the output response, and we plot selecting. At next, after the experimental design, the simulated response of the bridge we utilized to construct our.
Maybe we need to finish because the 10 minutes are already over. Would you mind talk in a few seconds?
Okay, ma'am, just 2 minutes. So we constructed our response surface models, and then we check the validity of our response surface model through R square parameter and RSME parameters, where we saw for R square average is 0.9, which is showing very good our good value, which shows that our constructed RS model is adequate. Then we constructed our objective function, which constructed our RS model, and the objective function is created such that the modal frequency of our RS model and the experimental modal frequency should be reduced, the difference between this, and then we used a global optimization technique for the optimizing this objective function. And after optimizing,
Stop it here because the next presenters are waiting. I'm so sorry for that.
okay, ma'am, just conclusion. Conclusion, we got this methodology used to develop a.
Speakers are waiting. I'm so sorry.
Okay, ma'am. Okay. Thank you, ma'am.
For your presentation. We will invite our next speaker from IIT BHU, India, Dr. Mahendra Kumar Pal. He will be giving a speech on application.
I'll share the entire screen. I did much better job.
Structures.
Hello? Hello, am I audible?
Yes, you are audible.
Thank you so much. My voice might, I might have a very tricky internet today as I'm traveling. So thank you so much for the nice introduction. And I'll directly jump to the presentation. Uh, Although the topic says that the application of high performance computing for fire safety of reinforced concrete structures, although the topic says the application, however, I'm basically going to talk about the fundamental development of a numerical technique, which in being implemented in the high performance computing environment, and 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 then how and what future we are is lying behind or lying ahead in this particular topic. That's what it is. So in the talk, I'm going to talk about the introduction methodology and some numerical verification of that, or means some numerical examples which I'm going to demonstrate to you. And with that, I'll conclude the presentation and then point it out what the future work which I'll be targeting or will be doing as a faculty at the IIT for that matter. So not going or not trying to criticize what is missing in terms of a fire safety in India, and specifically now when I'm staying in India, and making my salary out of here. However, what is happening here, not only in India, in the most of the developing countries, there is a lack of regulations, rather guidelines for against the fire safety. And most of you might have heard in your neighborhood, here or there, that n number of people are dying, or they uh, get hospitalized, or there is a lot of economical loss which is taking place due to uh, the fire hazards which are taking place. And particularly if you look into the countries where there are the cases of a fire in the forest and those kind of things, in those conditions the houses which come in contact with that fire, those comes in the become culpable of endangering the people's life who are living into that. And in that conditions, what it is essential that we should know, unlike other natural hazards like a seismic earthquake or the flooding or any other kind of a loading like blasting or anything, fire is another loading which has to be looked upon, and the guidelines has to be created for that matter. In order to create the guidelines, what it is important is that you need some sort of a numerical method so that you can know and understand how your building is going to perform, or how it is going to respond to the fire event, depending upon what is the scale or the intensity of the fire it is. And why I'm saying that numerical simulation, not the experiment, because as we all of know that fire can go uncontrollable, and therefore doing the experiment at the large scale with the fire will be like playing with the fire, and then we may damage the building in which we are doing the experiment itself, and that will give much better economical loss, as the quality of the people who are working into that particular building. That means the researchers are in much danger position, and we cannot put our thinker or the policy makers in that danger position. That's why what you have to focus, or the only option which is remaining, not the only option, but is the safest option which is remaining, is the numerical simulation. So basically, in this particular research work, what we are focusing is that we are focusing is the development of numerical method for modeling thermal cracking in the starting with the brittle material, assuming that because your concrete is the brittle material. And not only that, in the houses we have the ceramic utensils. We have a lot of antique items for a decoration in the houses, and those are most of them are manufactured using the ceramic materials, and that's where those are the brittle material, and we have to understand how those are going to fail, or how the cracking will happen and take place. That's it. And then after developing that, you have to validate and verify the numerical technique. And then I'm going to demonstrate how the thermal cracking the concrete, which we have done. We have simulated what has been done as the experiment by the James. So what methodology we are using is we are using the particle discretization scheme, which was my topic when I was a PhD student. back like 5-6 years ago. In the particle discretization scheme, what we are doing is that we approximate the function as well as a derivative over the conjugate domains. It becomes more or less a very mathematical thing, but let me put it in a simple way is that, for example, if I have to approximate a sinusoidal function, then what I can do is that I can simply divide it into n number of regions, and then go into that one particular local region, and then approximate my function in that particular one particular element. And then similarly we can do another the derivative of that also. That means the gradient of that I can approximate over the conjugate domain. So what basically we are trying to do here is that we are discretizing the entire domain. Or let's suppose that if there is a ball, or let's say there is a brick which we are want to approximate. So what we can do is we can divide it into the small particles, and then connect that with the usage of the springs, so that it will give me the entire body. That's what it is. The first minimizing the error. So one is known function.
We cannot hear you.
Benchmark problem what we did is there was the one experiment which was done on the aluminum oxide. So one plate of aluminum oxide was deep into the high temperature, 400 degrees centigrade and 500 and so on, and then it was cooled to 20 degrees centigrade by using the water bath. We simulated that particular experiment, and here
Mahendra, Mahendra, your
Yes, sir.
slides are not shared. Please reshare.
Okay. Thank you, Hasim Shah. Is it visible now?
Yes.
It's okay. So this aluminum plate, we have a model under the temperature 300 degrees centigrade, 400 and 500 and 600. As you can see that the white color, the gray color are the experiment results, and the blue colors pick slides are the basically the numerical experiment and what we have got from our numerical tool. If you see the cracking pattern in the both of these pictures, and then you can see that from the naked eyes, you can clearly see that the cracking pattern is as identical to what they have observed into the experiment. However, going into more deep into that, what we did is that we did the statistical analysis for that. That is missing. So that particular slide is missing. So what is happening is that here only the difference what we obtained was the length of the cracks which we are getting into numerical simulations are little larger than what it is in the what was observed into the experiment, and that is the reason there is a because the material properties of these materials are basically temperature dependent, and what we are assuming right now is that we are assuming the constant material properties. So once we update the temperature dependent material properties, then we might get or improve the results as in the future. That's what we are expecting. In the future, then in the next example, what we started doing was we took the thermal cracking of a concrete. So we take the one reinforced concrete beam, and like this, and then here you can see the red line. So along this red line, we imposed. There was experiment done performed by the Jamstick, and there the people they put the laser along this line, and then did how the cracking will take place in this kind of a scenario. So this is the results. As you can see here, so this is the beam. Here are the reinforcement bars are here. As you can see here, these are the reinforcement bars, and then after that here on the bottom there was the the laser beam was there. So if you see the results, so here we have the cracking which is seen in the numerical simulation results with the green color, and here you can see all the cracks are taking place. So from the naked eyes, what if you compare the results simulation and experiment, we have the cracking pattern is more or less identical to each other. However, if I go into the more detail, then there is definitely some discrepancy is happening. Even if you see the other side of the the other side of the same sample, here also here you can see that there is a very deep crack which is along the beam length, and here also you can see that there is a cracking pattern or the lot of cracks which has been generated along in the simulation results as well. So so far we have developed the tool in this much, and now what we are trying to do in the future is that we are trying to expand this to, right now we are just done for one beam element, but in the future we want to insert more columns into it and the slabs, and so that it will give the entire RC structures, and from there we'll be doing the simulation for the fire simulation of RC structures against fire safety. And for since the the specimen model or the numerical model will become larger and larger, and then I will need a supercomputer to perform the analysis, and for that we'll be using the Param Sivai, which is the first supercomputer in which has been recently established by the government of India at the ITBH. So with that, I'll end my presentation. Thank you so much. If you have a question, please you may ask or just write to me. Yeah. Thank you.
So much for giving a nice, wonderful talk, and we will.
सब कुछ correct. I am going to leave it. चलो सर.
Okay, thank you so much. Thank you so much, Mahendra-san, and welcome to Dr. P.K. Hashim from Hokkaido University. Today he will be giving a talk on nanoparticle functionalized with biomolecular machines. So, please, Dr. Hashim.
Okay, thank you very much, Chairman, and also Sunil Kaul and the Kedar for the invitation. I hope you can see my scene and you can hear my voice. Today I'm going to talk about the nanoparticle functionalized with biomolecular machine. So before going into the details, let me give you a brief overview of what's going on with me in terms of the scientific research project. The top one over there is completely related to the organic synthesis, and the middle one is something known for polymer-based drug delivery vectors for, especially for RNA. And the bottom here is for the biomolecular machine functionalized nanoparticle and corresponding hierarchical nanostructures. So due to the interest of time, today I'm going to talk only about the nanoparticle functionalized with the biomolecular machine. So everybody know nanoparticle has a core with the physical properties. For instance, the surface plasmon resonance can be tuned, and its surface have several ligands which can be tuned based on the ligands properties. So if we have a proper ligands, we can expect to encapsulate the guest, or we can even use for the drug delivery or for environmental sensing application. People have tried to functionalize several molecular level machines on the nanoparticle, as you can see here, or even the famous work from the Nobel laureate Sferinga, like the molecular motor has been attached on the nanoparticle. Even after attaching on the nanoparticle, the motor action is still active for UV and the thermal back relaxation process. So what I was wondering is whether we can attach a biomolecular machine on the nanoparticle or not. So I choose something called GROW-EL as a biomolecular machine. In nature, its function is to encapsulate denatured gust, refold it, and release in the presence of ATP. And this can be available, most of the bacteria, and also some of the variants of this GROW-EL is even available in the human body. So once again, this is a biomolecular machine, and its function is to encapsulate denatured gas, refold it, and release in the presence of ATP. So two properties of biomolecular machine of GROW-EL is hollow structure to encapsulate the gas, and the second is ATP responsiveness. So to give you the structure of this protein is something like this. It's an heptamer of protein subunits that are stacked back to back, and it has a core cavity of around 4.5 nanometer, as you can see here. And the most importantly, it can respond to ATP even in the isolated form, not only in the biological GROW-EL. Even after isolating with the bacterial expression, this GROW-EL is still active for the GROW-EL induced properties, for instance, guest encapsulation or ATP responsiveness. So this is the model which I really wanted to construct is GROW-EL. This is the scheme that is fully functionalized onto the surface of nanoparticle. So I used the strategy of DNA hybridization, where the DNA is first attached on the both side of GROW-EL protein via a cysteine mutated protein with a Michael reaction. And then the complementary DNA is attached on the surface of nanoparticle. Now, simply mixing and thermal annealing, what you can expect is this DNA can hybridize and prepare this kind of GROW-EL functionalized nanoparticle. Of course, for ensuring fully functionalization, you need a lot of GROW-EL. So we use about 100 times excess of DNA GROW-EL with respect to the DNA nanoparticle, and then thermal anneal it, and to get the mixture of unattached GROW-EL and the GROW-EL appended nanoparticle. First, we confirm this via solution-based methods, something known as DLS, dynamic light scattering, where you can see the size of nanoparticle appended with the GROW-EL. And also you can see the size of GROW-EL. Both are in less than the size of the conjugate. So you can differentiate whether the structure is formed or not. After mixing and thermal annealing, you can see here something large structure around 92 nanometer form. Correspondingly, the zeta potential that check the surface properties of the conjugate, you can see a marginal increase of negative charge of the protein on the nanoparticle, indicating this kind of structure is formed. And as I said, I use the GROW-EL as 100 times excess, so I need to purify. So we use something known as sucrose density gradient, where you use a different layer of sucrose on a tube and then apply the centrifuge. Now you can see that based on the sucrose concentration, you can prepare the different layer. The heavy material, I mean, compared to the GROW-EL protein, the GROW-EL appended nanoparticle moves faster. So as you can see from the color, then you can cut this band or you can isolate this band to get the pure form of the GROW-EL protein appended nanoparticle. To confirm, we use the TEM, transmission electron microscopy. This is a cross-sectional image of the three-dimensional structure. As you can imagine, the original structure is 3D, but the image is 2D. So you cannot see the GROW-EL on the upper part and also the bottom part due to the large electronic transmission of the nanoparticle. But if you look at the periphery, clearly the GROW-EL, the protein, the kind of structure can be seen. Even you can count the number of GROW-EL per conjugate. And if you make the histogram, it's something like this. So this histogram shows the number of visible GROW-EL per nanoparticle. And then if you assume one above and one below the conjugate, then this would be exactly expected from the theoretical modeling study. So in a maximum, we can see 7 to 18 GROW-EL per nanoparticle. Now, this is a beautiful structure, but what you can do with this? You can do several things. Number one, for instance, although GROW-EL, this biomolecular machine, is attached on the nanoparticle, it still functions as the original biomolecular machine, which means you can encapsulate the gas and release in the presence of ATP. And also, we serendipitously found the conjugate shows higher ATP hydrolysis enzymatic activity than the individual GROW-EL. From a structural perspective, you can also extend the layer of GROW-EL, number of layer on the conjugate, even to construct the hierarchical nanostructures. So let me briefly tell about how you can show the layer extension. So as I just talked, by complementary choosing the DNA and its sequence, and then thermal annealing, you can prepare this and purify a GROW-EL appended nanoparticle. And then by using a different complementary into the complementary of the first strand, then you can prepare the layer extension. So one problem here could be like the sequence on both sides are same. So when you do the second layer extension, there is a possibility that the first layer is ripped off. To avoid this, I use something known as the glycerol cushion, again in the annealing mixture, where you can control the annealing temperature to avoid the ripping of first layer and to construct the second layer on the GROW-EL. So what's the proof for whether this two layer is formed or not? Again, in the transmission electron microscopy, you can see clearly here the second layer of GROW-EL is attached on the first layer. And from a quantitative analysis, we found that each of the GROW-EL layer on the nanoparticle in total have around 10 GROW-EL per nanoparticle, which indicate all of the GROW-EL on the first layer is iteratively extended into the second layer. And then if you want to construct the third layer, fourth layer, in principle, it is possible. You simply need to care about the thermal annealing temperature so that you shouldn't rip off the previous layer when you make the third layer. Based on this concept, if you want, you can prepare a star kind of a polymeric layer on the nanoparticle by using our method of using a cold rigid protein and using the proper DNA hybridization strategy. Now, this is about the layer extension. The other property is about the ATP hydrolysis. As I mentioned in the first part, this is a biomolecular machine, so it has the ATP hydrolysis activity, which simply means to convert ATP, that is adenosine triphosphate, into ADP, adenosine diphosphate. And this we serendipitously found on grow eel, attaching on the nanoparticle shows very high, about 5 times higher enzymatic activity compared to the free grow eel or DNA appended grow eel. You can see this graph shows about 5 times higher. This, we don't know exactly why this is happening, but somehow this is happening, and it's been confirmed by the reproducibility of the experimental result. And as I said, even after attaching grow eel onto the nanoparticle, it still can encapsulate the gas. This is a model study where we use the GFP, that is green fluorescence protein, and we denaturate it and then mix with the system, and then we incubate it for a certain time to refold the GFP. So this graph shows the emergence of fluorescence that indicate the refolding activity of GFP on the time. You can see a clear increase in the fluorescence intensity, indicating that the refolding activity is maintained even after attaching onto the grow eel, onto the nanoparticle, sorry. So now, like from a nanostructural point of view, what you can do is like you can construct the hierarchical nanostructure based on this concept. There is 2 ways. One is the one-pot assembly. You precisely choose the DNA hybridization motif and then mix it together and incubate it for a certain time. You can construct this kind of porous nanorobotics cluster. Why nanorobotics? Because each linker can move based on the ATP. So this will be kind of a robotic action can be expected in the presence of ATP if we have this kind of nano-based structure. If you construct on a stepwise base, like as we just talked now, let's first construct the grow eel appended nanoparticle and then use DNA as a linker. Now the pore size in this cluster is much, much larger than this one. So due to interest of time, I will briefly show the experimental result from the one-pot assembly. So here is the TEM image from the one-pot assembly of a smaller nanoparticle. So what I talked before is the 20 nanometer one. Now is the 5 nanometer one. And you can see that clearly each nanoparticle are well separated via a grow eel linker. This indicates a nanorobotic cluster is indeed formed in the molecular level. And other properties, something like the ATP-induced motion, all are now currently undergoing. So in summary, what we achieved is an ATP-responsive nanoparticle, functional as the biomolecular machine, which can encapsulate the guest and release in the presence of ATP. From a structural perspective, this can use for layer extension or even for the nanorobotic cluster. And serendipitously, it shows that we found that the conjugate with the grow eel or nanoparticle shows 5 times higher ATP hydrolysis than the corresponding grow eel. And before concluding my talk, I want to acknowledge my previous mentor, Takusho Aida from the University of Tokyo, and the student colleagues who actually did the research work. And finally, the funding from Concert Japan, as well as the JSPS company. And thank you all for your kind attention. Thank you very much.
Thank you, Dr. Rashid, for such a wonderful, outstanding talk. And it was nice to hear about the grow-eel linking, as well as the nanoparticles linking for the grow-eel. Thank you much. So we will start with our next speaker.
I hope you are I'm audible to you, and you can see my screen.
Let me introduce you.
Okay.
Our session is very multifaceted. Our next speaker is, after the brilliant talk on molecular machine, biomolecular machines, let's move to Dr. Sharad Srinivasamurthy. He is assistant professor at Institute of Ocean Energy, Saga University, and he will present his research work on wind turbines. Zooming over to you, please start.
Yeah, thank you. Thank you for the kind introduction. Am I audible now? Is it all right? And I hope you can see my slides.
Please start.
Yes, we can see and we can hear you.
Yeah, thank you. Thank you. Yeah, thank you for the kind introduction. So my name is Sharad, and today I'll be talking about my recent research outputs. It is related to numerical and experimental investigation related to the slow death damping, which we discovered for a single point moved floating offshore wind turbine. So before getting into the research topic, I would just like to have a brief introduction about our research institute. Our institute is called as Institute of Ocean Energy. It's in Saga University, Japan, and it's to the western part of Japan. And it's a research center which is dedicated to ocean renewable energy. And we work on elemental technological development and which to address global environmental issues. So basically, we try to provide solutions from to harness the umpteen ocean energy. Here in this figure, we can see the yes. So this is our experimental facility. It's in the Imari city in Saga Prefecture. Our organization is basically it started as in a humble background in 1973. Just a simple apparatus to convert ocean thermal energy, and now it has reached into various sectors of ocean energy. And we also have various collaborations and joint usage across the nation. The first department that we have is the Ocean Thermal Energy Department, under which there are 3 divisions, the Thermal Energy, OTEC, and the Innovation for Thermal Energy, and the third is the Deep Ocean Water. Ocean Fluid Energy Department is our second department, in which there is division of wave energy conversion, and the division of tidal and ocean energy, and division of offshore wind energy. So here we can see that the research started in 2017. So it's quite recent because offshore wind energy itself started, the research of wind energy itself is very recent. And I belong to the division of offshore wind energy, and it's an emerging technology. And I started my work in Saga University 6 months ago. And we also have interdisciplinary research department, which also we collaborate with the professors, and we collaborate and provide technological advancements in 5 different fields. First is the ocean environment. The second is the energy storage, especially related to the ocean energy storage through hydrogen, and ocean energy control systems, which is essential for most of the applications right now. And the material production, which are the byproducts of the ocean, like the lithium recovery, the aquaculture, and desalinated water. We also have collaborated with the human development education, because we need skills to be transferred to human skills. We need the human skills to understand the ocean in a better way. Of course, we all know that oceans cover 70% of the ocean, 70% of the earth, and we know very little about it. So overall, we are 29 researchers, 15 full-time faculty, 11 collaborating professors, and 3 postdoctoral researchers. So the location, as I mentioned, is in Saga Prefecture. We have 3 main campuses. One campus is in the main office, headquartered at Saga City, Onjo campus. The second one is the experimental facility at Imari Satellite Town, which was established in 2003. The third one is the Kumejima Satellite, established recently in 2014. It's an experimental facility where there's a 100 kilowatt plant, which is established. And it's a model for future OTEC plants. It's also interesting to note that this facility in Kumejima, it's one of its kind, and Okinawa, it has about, in this Kumejima town, there are about 8,500 people, and it may be sufficient to utilize just the ocean energy to survive, to cater the energy requirements of the island. We have major facilities, which include the OTEC related, about 6 devices. We also have the fluid related 4 devices, and the material recovery and the hydrogen related devices. So overall, we have about 22 devices, which also can be used for collaborative research. And we also published a technical journal every year. It's an annual report. It's called OTEC Annual Report. And these are some of the facilities. We have some international activities at IIS as well, with 3 main international events: the International Seminar on Ocean Energy, Young Researchers Forum on Ocean Energy, and International Platform on Ocean Energy for Young Researchers, which we had just last week. We have about 1,600 people visiting from 89 different countries, and all collaborated with IIS in the past 10 years. And we are really looking forward to collaborate with you if you are interested in ocean-related technologies. And we also provide free facility tours for interested researchers, even online. So please feel free to contact me later on. So now coming back to my research topic. So how much of ocean energy that we are really talking about? So there is a huge potential of ocean energy. And this slide, in this, we can see that wave energy, this is one of the predictions, the estimations that we have is 45,000 terawatt per hour. The ocean current is 400, tidal is 1,800 terawatt hour, and ocean thermal is 33,000. So these are in terawatt hour per year, in the osmotic energy as well. So ocean energy theoretically has the potential to satisfy the present global electricity demand on its own. And in this, we don't even consider the offshore wind energy potential in this study. So we have such a huge potential, but how much of it is actually used in the present situation? In the present generation, in the present case, we hardly use about 2% of the available energy that we have. And this 2% includes the wind, solar, biomass, geothermal, and ocean power. So we can see how heavily we are dependent on the fossil fuels for generating our electricity. Of course, everybody knows we need electricity in a clean way, but we don't have option other than until we reduce the cost to a certain limit so that it is competitive. And that's one of the main intentions for our institute, to reduce the cost. And in all these energy sectors, the wind energy is the front runner in the modern renewables, only related to the technology which has developed for years and established technologies. And onshore wind has various limitations because of the high-rise buildings, and there is not enough strong wind onshore. So that's one of the reasons why we are going offshore, as a wind resource is very strong and also untapped. So what is offshore wind energy? So we divide the offshore wind energy into 2 categories. The one is the fixed bottom, and other is the floating offshore wind turbines. So floating offshore wind turbines are emerging technologies, and the fixed bottom have been already almost established. So basically, a wind turbine consists of 3 blades, most common type of wind turbine, which has 3 blades rotating, and it's connected to the hub, and the nacelle generates the host generator, and everything is hosted on a tower, and the tower is supported on the land in the onshore case, and on the ocean we need to have a foundation to support this tower. And here we cannot see what is below the water, but actually because I know this is a floating type of wind turbine, so it has a floating structure called as a spar type. So what is the meaning of a spar type is this kind of spar structure. So for a water depth of 0 to 60 meters of depth, we use fixed bottom type turbines, fixed bottom type floating structures, fixed bottom structures like monopile or a jacket structure. But this will not be sufficient, will not be economical if we go deeper in the ocean, above 60 meters of depth. So we need to use floating structures. The floating, basically there are 3 main concepts of floating structures. The one is the tension deck platform. The second one is the spar buoy, which is used in this particular case of floating offshore wind turbine. And we also have a semi-submersible case concept. All these 3 concepts are basically having a floating structure. So we can expect 6 degrees of freedom motion, but they don't drift apart in the ocean because we use a mooring line to host them at a single location. So there are about 27,208 megawatt of power of Wicks bottom, which has been already installed, and 82 megawatt of floating, which is installed presently in the whole world. But since these are emerging technologies, there is high potential to reduce the cost in the floating sector. So basically, it's a very interdisciplinary engineering topic. It involves mechanical engineering, electrical engineering, the control engineering, structural engineering, as well as ocean engineering. So how we integrate and develop methodologies to come with an economic optimum is the ultimate challenge here. So to address this, we also developed frameworks for Japan related to the Japanese seas. So one of the most important thing for a wind turbine is to have a yaw control system. And for an offshore wind turbine, the maintenance becomes very costly and challenging if the yaw control system breaks down. So what is an alternative for it? The alternative is to use a single point mode floating offshore wind turbine system. So what exactly is a single point mode floating offshore wind turbine system? So if you have a wind turbine in this particular location, and the wind starts flowing from this direction, the floating offshore wind turbine can align in the direction of the wind by rotating in this particular direction. So that's basically a concept of single point mode floating offshore wind turbine. Here, if we use a downwind type wind turbine, we can generate electricity and assume if the wind comes from the different direction, it will align in the direction of the wind. And therefore, we can eliminate the yaw control completely, and we can save the cost, both in terms of the maintenance cost as well as construction cost, because it just uses single point mooring. So in this the research, the main objective was to study the influence of slow drift damping on the weather vane performance of a single point motor system. So this was the concept in Japan. This has been going on. This is another sponsored project in which we are trying to develop a framework to reduce the cost by 2030 to 20 yen per kilowatt hour. So this is the target, and this concept has to be verified, and this concept can be used to achieve at such economic costs. So as we see here, there are many challenges to have such kind of floating structure generating wind energy in the ocean. The first challenge is to weather vaning, because if there is it's moved at only one location, whether it really aligns with the wind direction is the first question. Then we also have to understand the current and the wind loadings, and we need to understand whether it's stable. There is also a problem of phenomena of slow drift damp motion, which happens for such kind of single point mode structures. So the research methodology we adopted was firstly to conduct small scale experiments, and then we move on towards understanding the mechanisms using numerical simulation. So to understand the complex energy systems, we need to explore new concepts, and this is one of the new concepts. So in the experimental model, the experimental models of 160 scale was used, and it was conducted in National Maritime Research Institute in Tokyo. And here we use a ducted fan, which is a new concept for simulating the thrust force occurring at the tower top. So if we call it as a real-time hybrid testing model, because in real time, we can understand the wind loads, and we can avoid the Froude and Reynolds scaling function conflict by using this kind of a testing technology. And this is the first time that this technology is used on a single point mode floating offshore wind turbine. And we evaluated the weather vane performance and understood the stability components. This table shows the displacement. That's basically the weight of the structure. So it's about 31 kilograms. So you can imagine in real scale, this will be somewhere around 6,700 tons. So these are mega structures. So this tower, which is about, in the real scale, will be, you can imagine it's somewhere around 80 meters. So you can imagine how huge these structures are. We used motion capture cameras to track the motion response. And this was the experimental setup that we carried out. It was moved, this is the model. And we moved the model at one location using 3 cables. and then we start the wave from this direction, because it's a wave making device from this direction, and the wave comes in this direction, and we also generate the wind in the same direction. And then we evaluated how the motion and the response of the structure is by conducting various changes in the direction of the wind, as well as the duration and the wave height and the wave frequencies, and also the current speed. So we varied all these things and came up with an idea that how this particular concept can work. So in the numerical modeling, it's with a very simple numerical modeling explanation here. We try to develop the 3 degree of freedom motion in the surge, sway, and yaw directions, as shown in this figure here. And the external loads that are coming onto the structure is basically the wind load, the current load, and the mooring load. So since this slow drift damping motion is the most important parameter, because the yaw direction it has to move. So we conducted some kind of an experiment to evaluate and obtain slow drift damping coefficient P by using such kind of a post oscillation device. And the idea is to use this this values obtained from this experiment in our numerical simulations, and we used Runge-Kutagill method for numerical simulation. Oh, yeah. So finally, so this is basically the results that we obtained. That when we have the simulation results with the slow drift damping and without the slow drift damping, when the simulation which consists of experiment with the slow drift damping compared the experiment results quite finely, and without the slow drift damping, the simulation cannot predict the experimental results. So this is one of the latest findings that we understood, and this was for the case of rated wind, the cutout wind speed, and the storm wind conditions. So slow drift damping coefficient was found to be a very important parameter, and it needs to be evaluated for single point motor mode floating offshore wind turbines. And the new methodology is also validated, and it can be used for other single point mode floating structures. So the more details of the study can be found in this paper. Thank you very much.
Thank you so much for this wonderful talk and letting us know about your institute and your research and the potential and how to solve the wind turbine energy and use effectively. So let us move to our next speaker, Dr. Paresh Halkar. He is also from the same institute. Institute of Ocean Energy, Saga University. He is currently working on floating body pendulum wave energy converter. Now, zooming over to you, Dr. Paresh. Please start.
Good afternoon, sir. Is it audible?
Yes, sir.
Yes, sir. Is it audible?
Yes, it is.
Thank you. Thank you so much. My title is basically experiment numerical analysis on wave energy converter using ocean breaking wave. This is my outline of my presentation. First, I'll talk about the small introduction on different type of energy converter and wave power in the world as well as India and Japan. And this purpose of my research, I have to discuss small regarding that concept, turbine design, lab experiment, and finally real sea experiment and summary. Yosom Masuda may be the regret that further of modern wave energy technology with studies in Japan since 1940, he developed fast navigable buoyancy powered by oscillating water column. At the present, that research and development of the world concerning that wave energy converter is very attractive because the recent high oil price and global warming. Wave energy converters developed projects are now various phase of the study across the world. Why that wave is important? Accordingly, 2017 new record the energy related CO2 emission 33.14 gigaton plus 1.7 increase. Accordingly, wave density design that only wave installed 0.006 gigawatt per hour, almost 0.03%, while the power density is much higher than that wind as well as solar. How that wave energy is working? That initially the wave is shallow water or near shore or offshore. Then it is captured the wind energy capture mechanism, and finally that used for turbine by power take-off mechanism. Finally, we'll get that electricity. This is different kind of wave energy converter. That one is the power take-off hydraulic motor, hydraulic turbine, linear electric generator. ISAJ heaving effect means vertical direction, heaving and pitching, vertical and rolling, and pitching only the rolling. And oscillating water column basically is a PTO system used for impulse turbine and Wells turbine. And this kind of basically two. One is fixed oscillating water column, this onshore, and floating, it is offshore. Over another PTO system, basically overtrapping wave energy converter. Wave power, annual wave power. This is basically prevailing western side, means that the wind's latitude is 35 to 65 degrees, where that wind force is very high. The wave power is incident wave calculated by 0.5 in significant wave height square multiplied by significant wave period. According to average wave power around the Japan, 7 kilowatt per meter, where is Indian average wave power is around the coastal area in India 5 to 10 kilowatt per meter. Purpose of this research to generate the wave energy using breaking wave, first proposed by Professor T. Shintake, Okinawa Institute of Science and Technology in Japan. To find the numerical analysis that can evaluate the power coefficient, generate and turbine running characteristic of the device. Concept of this design, that average when that wave is approached towards when the approach conversion possible when the approaching shore steepens due to the decrease the water depth and wave energy condensation near the surface. The steepened wave can be reached 4 to 6 meters per shore head before it breaks in the surface region. So in this case, the rotating blade take the advantage of breaking phenomena in the surface zone to harness the wave energy into electricity. So blade design in the figure that y-axis power efficiency and x-axis tip pitch ratio. If low TSR, that number of blade 5. When increasing the TSR, the number of blade is decreases. But in this case, we need that number of blade is higher. We require this number of blade is higher. So in this case, why low gear, low twist ratio, that high acceleration and low impact. Twist angle and chord length. The twist angle calculated based on that tan theta VF by R velocity and omega angular velocity multiplied by radius. And chord length uses following this equation Wn, CL is lift coefficient. In the present case, lift coefficient we consider the 0.3 and efficiency is also considered 0.30%. Accordingly, that near the half section, that the TSR is higher. When increasing that tip section, the TSR is low. That optimum chord length decreasing with increasing the radius. The experiment carried out fundamental research in wave energy conversion, such as inlet constant velocity and conversion power coefficient. The laboratory experiments carried out measured West Japan Fluid Dynamics Laboratory. That in this case, that only half scale turbine used. The experiment results power coefficients Cp equals to torque into angular velocity and rotational speed by rho p cube means velocity in surface area. Turbine project surface area. Tip speed ratio omega by V0. Velocity is range is 1 to 3 meter per second. Based on experiment, optimum velocity 2.5 meters second. There is different inlet velocity used in for experiment 1.5 up to 2.7. That optimum velocity is considered 2.5, where maximum power around 0.25. These generator characteristics, the generator characteristics based on laboratory experiment, the different load, input load 4.7 to 13.5, the load is increases, that power decreases. While electric power, when load is decrease, that power also decreases. With increasing load, power decreases. Comparison, numerical and experimental results for power coefficient. This is CFD simulation. Computational, this is similar. Consider the same as experiment model. The rotor, blade, hub, and blade imply no slip conditions. Local averaging rotating domain used for variable angular velocity, and then constant velocity profile used at the inlet. These experiment results, experiment and numerical results are compared with that. It shows that the numerical is well agreement with the experiment, and the average deviation CFD and experiment only 9%. The CFD results, the CFD results is considered a different inlet velocity with different rotational speed, and we draw this figure. We'll get that increasing the inlet velocity, that power also increases, while also drag in the next, the drag force also increases with increasing the inlet velocity. Now, this correlation between generator turbine is CFD. We got that CFD turbine power output based on the CFD analysis, and generator power output based on the experiment results. Now, we fitted this curve with power versus RPM. We'll get that corresponding cross point. Then we extract that point and create that another plot. So we'll get that mechanical load versus electrical load. And in this case, that maximum mechanical load is considered 30.4.9 and minimum mechanical load 4.7. Now, after extracting this all the point, we can create that variable response, like mechanical load, inlet velocity versus RPM. If inlet velocity is increases, what is the RPM? Similarly, inlet velocity versus drag force. If the inlet velocity increases, how much drag force will create? And similar as the how much power can be based on that inlet velocity. This is the one table. If inlet velocity 1, that turbine RPM 0, power 0, and that minimum inlet velocity if 3, the maximum RPM, that RPM can get 100, while power output 75. If increasing the inlet velocity 7, the maximum power output 12 kilowatt, while the rotational speed 74, and the torque and corresponding the drag force. It based on that mechanical load low, 4.7 ohm. This is a real experiment of the energy at the Maldives. The wave energy experiment sites near the in Maldives, such Kandama Island in Maldives. This is a this is a test site and half-scale unit installation near this island, and there is 2 kind of unit is installed: WC1 and wave energy converter 1 and wave energy converter 2, and difference between 2.5 meter based on that high tide and low tide. These are 2 half scale installed. One is this one, and this is another one. And later, that full scale turbine installed in the same place, and that height same. It is installed in 2009 in July, and this is connected through cable connection bus and wave energy monitoring system besides the island, generator plant house, and Kandam Island in Manali. This is the wave monitoring display. We are getting that value using the remote desktop. That the voltage is maximum 300 volt. That upper full scale WC3 AC voltage versus time around at time 0.3 second, output voltage 315 volt peak, and magnifying that output voltage 0.224 seconds, 0.4 seconds, the frequency 20 hertz, while rotational speed 300 RPM. The summary, it shows that the numerical results are well agree with the experiment results. It is observed that maximum CP 3.8 while TSRN 2.5. It can be seen that impedance is the critical factor in determining the turbine starting characteristics. The running characteristics of the turbine is controlled by the wave frequency and incoming wave velocity. Thank you so much.
Thank you for this wonderful presentation, Dr. Paresh Halder. If anyone has any questions, please write in the chat box. And then let's move on to our next. After listening to these two talks on natural sources of energy, let's welcome our next speaker, Dr. Hemant Muthalapati. Let me introduce him shortly. He moved to he joined Shimane University, Japan in 2014 as an assistant professor at Raman Center for Medical and Biological Applications. Then onwards, he has been involved in various responsible roles. Currently, he is also associated with the Faculty of Life and Environmental Sciences. His scientific research is primarily oriented towards developing early diagnostic procedures for cancer and other diseases using artificial intelligence-assisted Raman spectroscopy. Additionally, his efforts are focused towards university research strategy, internationalization of higher education in Japan, creating a strong network between premier educational and research institutes in India and Japan, and enhancing Indo-Japan cultural exchanges. And now zooming over to Dr. Muthalapati, please, the stage is yours.
Thank you very much for the introduction. So I am Hemant. I am an assistant professor in Shimane University, and this is my contact information if you want it's here. And what I'm going to talk about today is accurate classification of breast cancer cells from normal breast cells by machine learning assisted Raman spectroscopy. So what I'll do, because the audience are coming from very different backgrounds, so I'll just briefly explain what is Raman spectroscopy, what is the principle, and how do we do the experiment, and then I'll just jump into the results and show how it is very useful. So what is Raman spectroscopy? Raman spectroscopy is just an analytical technique based on the light scattering, and what we are primarily collecting is inelastically scattered lights. So in order to understand why it can be very useful, as I said, this is a technique that is trying to probe each molecule, right? So this is just a CH2 moiety, for example, a part of the molecule, and you can see that there are lots of different kinds of vibrations within each molecule. These are intramolecular vibrations. Of course, there are so many different kinds of vibrations, like stretching, scissoring, rocking, and so on. And each of these vibrations has certain amount of energy associated with it, or because of the frequency that's involved here. So we can actually use this energy to actually identify what kind of vibration it is. And by looking at the nature of vibration, you'll be able to actually identify what kind of molecule it is. So this is just an example of one simple vibration, but when we put together all kinds of vibrations together, then we'll be able to identify each of the molecules. And it was actually discovered by Sir C.V. Raman in 1928. And in 1930, just 2 years after the discovery, he got a Nobel Prize. So that already shows how important the technique is, because within 2 years of discovery, he was awarded Nobel Prize. So let's see what are the different kind of light scattering processes that exist, and then how do we measure them. For example, this is just energy diagram. let's say there is a molecule in a ground state, and if, for example, I'm going to shine some light of very specific frequency, for example, say using a laser, then what happens is it's going to go to some kind of a virtual state. There are much stable states over there, but this is some kind of virtual state where the molecule cannot really stay there, so it immediately comes back. So the main point here is that when it comes back to the same state as it started, then there is no energy difference. That means there is no either gain or loss of energy from the excited photon. However, so this is actually the most common scattering that we always see in our daily life. This is it's very elastic scattering. There's no exchange of energy. However, very tiny percentage. For example, 1 in a million photons that interact with these molecules can actually, gain or lose energy. For example, in this case, this is a Stokes scattering where the molecule started from the lowest vibration energy state. While coming back, it goes to a higher vibrational state, ground state. So that means there is some kind of a difference in this energy, right? For example, from E0 to E0 plus H nu m. So this is the difference, right? This difference is actually what is shown here. So difference in energy means difference in frequency or wavelength, eventually meaning difference in color, to simply understand. So when we shine with green light, we get red scattered light. That means it lost some energy, right? Similarly, on the other way, if some molecules are already in a vibrationally excited state, it can also gain energy. So when you shine with a green laser or a green light, you may also, 1 in a million photons may also actually gain energy and turn to blue. So if we can actually collect these photons that have changed color from the excited photon, then we can see the energy difference. So this difference basically means this difference. That means this difference is associated with the frequency or the vibration, intramolecular vibration itself. So that's how I think we can imagine. Let's see, how do we do experiment? For example, this is a very simple model where, for example, there is some kind of chemical. It could be the solid, liquid, gas, any phase, doesn't matter. So we are going to shine some laser. It's a very strong, say, green laser. You can see most of the scattered photons are all green, but however, we can see some other colors coming out. So if you see what the green is very, very strong. So what we can do is just simply cut it out and collect only the photons that have, for example, say longer wavelength. Then instead of, because these are very, very few photons, so we cannot actually see with our eyes, impossible. So what we do is we use some kind of a highly sensitive CCD cameras, and we can actually generate this kind of a nice Raman spectrum. And this is Raman spectrum in this range, wave number range. And the most important thing that we must understand is that each Raman spectrum, this is a vibrational spectrum. So that means this is some kind of a molecular fingerprint, just like what we have. Our fingerprints is unique to each one of us. So each molecule has its own vibrational fingerprint. So that is the main advantage that we can, why we can identify molecules. So this is just a schematic of what I built in our laboratory. So we just couple that with a microscope, then we can actually study microscopic sized samples. Primarily what we are looking at are cells, tissues, and so on in our laboratory. So this is how the actual spectrometer or microspectrometer looks like. The advantages are that we can do actually in vivo experiments. I mean, because it doesn't really affect or cause any damage. So we can also say it is sometimes minimally invasive or sometimes non-invasive measurements are possible. And we can actually look at obtain space and time resolved information with sub-micrometer resolution. And one of the biggest advantages is that in the whole process, we do not do anything else to the sample. There are no sample pre-treatment. In order to actually probe the molecules, we do not use any other dyes or fluorescent dyes or anything. So we just look at it, look at any sample as it is. And in fact, as I said, when we combine with the microscope, what we can do is we can also do imaging of micro-sized particles or samples. So let's assume that this is a cell, say for example, 10 microns in diameter. Then if we just, for discussion's sake, we just make it in 16 points. And if you just shine laser at this particular point, we get some Raman spectrum. For example, is outside the cell, there is nothing here. But as you actually scan through the whole sample, you can actually generate such series of Raman spectra, which can be tagged to certain position or space or within the cell. Then what happens is each of this, for example, here I'm showing only between 1400 to 1800. So there, usually we take thousands of these spectra, okay, for each of the samples. Then if you put all of them together like that, actually in each spectrum there are about 1300 points. And then eventually if you have some, say, in this data set that I'm showing right here, it has about 8.5 million data points. So this is actually kind of big data. And we do lots of different kinds of data analysis, and then we can generate pure molecular components, images. So we can get this to molecular images without actually doing anything except for shining light. So that is the background of what is Raman spectroscopy and what can be achieved using Raman spectroscopy and microscopy. And for today's discussion, what I'm going to talk about is primarily the application of Raman spectroscopy for breast cancer diagnosis. In that sense, we wanted to first discriminate which are breast cancer cells and normal breast cells. In fact, this topic is not new. It's been done for a very long time. People have done it, using Raman spectroscopy combined with PCA by actually, trying to discriminate breast cancer cells from leukemia cells, like blood cancer cells. And they also, some have done all sorts of things. But the main problem with this is each of them have their own approach, and the most important point that the spectrum carries molecular information. They do not, none of them actually discusses why we can actually discriminate, or whether it is reliable or not, and so on. And honestly speaking, by if you have one Raman spectrometer, I have another Raman spectrometer, the performance is different. There are so many parameters that have to be tuned, and in order to actually compare data sets. So that makes things very, very complicated. In fact, that is one of the reasons why it's not translated to clinics yet. However, if we are able to actually make discrimination based on pure molecular information, then it would be very easy for us to actually transform this technique to clinics. So that is why that is the main motivation for my research. I'm sorry. So this is a very simple experiment. We are just measuring two different kinds of cells. One is normal HMEPC, human mammary epithelial cells, and the other is a breast cancer cell. And it's color coded, so it's either blue or red. And red means cancer. And we are measuring lots of cells, and then we are trying to see what happens. So in the case of, if you just take an average Raman spectrum of, say, normal cell and the breast cancer cell, it looks more or less similar, kind of, except for some experts in the field, you cannot probably you can say that there are some differences here or there's some difference here, but this, mind you, is an average spectra. These are average spectra. So it is not very easy to actually make any decision with this. So if you still want to use some traditional method and make some kind of to show that there are differences, yes, we can do that. However, we cannot really pinpoint to any single molecule, because each of these Raman spectrum here is actually a mixture of all kinds of molecules at that position. So we cannot really make any concrete discussion about the molecular origin. But anyways, in order to actually see whether this data set actually whether we are able to actually discriminate these two kinds of cells using the data set, the first very simple, straightforward multivariate analysis that we use is principal component analysis. And within the first few components, we already close to about 92% of the data are represented within 4 components. And the first few components can actually make very nice distinction. And this is kind of exploratory analysis. So what we do is we can further go and use supervised machine learning methods, including SVM and LDA and so on. And here what we do is we do some kind of training, and then we try to actually use this for prediction. And in fact, we can actually get very high accuracy using applying linear discriminant analysis on the first 4-5 PC components. And again, in order to actually see without any much of human interference, we also wanted to try, say, machine learning for example, say SVM, and we also get very, very high accuracy. But the problem again here is that it is very severely limited in two aspects. One is that these do not really give us any molecular information. So that means we do not know on what basis it's actually separating. That means we cannot completely, transport it to in another situation with another data from another machine and so on. It cannot be universalized. And another point is that this does not actually take into account lots of other factors. For example, depending on the nature, depending on the nature of the cell or where you measure about the sample. I'm not talking about the experimental parameters, but I'm talking about the sample itself. And so these two things have to be addressed. So the first one is trying to understand the molecular information so that we can make reliable prediction is done by using MCR analysis, where we this is one of the very special things that we do in my laboratory, where we try to extract pure molecular information from these complex data sets so that we can make physically interpretable spectra. So simply we have some kind of large data matrix. What we are trying to do is we try to decompose into two different matrices, one with pure spectra and the other with the corresponding abundance profiles and so on. But while doing this, what we do is that we actually apply some constraints, especially some non-negativity constraints. So because Raman spectrum cannot be negative. If you see in case of PCA, the PC loading spectra, even though it looks like this, the problem here is that these are all, it contains both positive and negative features, and these are not pure components. These are actually linear, multiple contributions are there. And in order to actually obtain pure components, we may have to do some kind of linear combinations here. But as you increase the number of components, this simple linear combination may not be very straightforward in this case. That is why what we do is we use alternating least squares in order to actually fit the data. And by using such constraints, what we get is actually pure physically interpretable Raman spectra. Let's see. From this data, of course, by actually looking into what are the number of components that can take so much of data into account, so we decided that we construct a 7 components model. The one thing or the limitation of this model is that the model itself is actually, we have to, the number 7 here, for example, is an user input. Like we have to decide. And there are so many ways that we try to identify what would be the best model. Anyways, we constructed a 7 components model, and out of the 7 components, 2 are background spectra, but the 5 beautiful Raman spectra that we can see, these are pure molecular spectra, unlike other analysis. And out of that, we actually identified 3 independent lipid molecules. So that is wonderful because even though the nature of lipids are very, very similar, the molecular structure itself varies in terms of, various, for example, the double bonds and so on. So we can actually extract or separate even such detailed molecular information using our analysis. And the corresponding Raman abundances or the concentrations are given in this case. This is for the normal and this is for the cancer cells. And if you actually go on and do have some kind of analysis interpretation, what we can see is that this lipid 1 actually makes very, very clean discrimination, the relative ratio of lipid 1 to, say, nucleic acid. So I said there are 5 components. 2 of them are one of them is nucleic acid, the other is protein, and 3 of them are lipids. And we still haven't really discussed what kind of lipids they are. I'll come to that in a bit. But before that, what I wanted to show you here is that the relative ratios of lipids, especially lipid 1, to nucleic acids or even other lipids, can give very, very statistically significant difference. Well, what are these 3 lipids? So as we see, these 3 lipids have this band. This band is actually coming from CC double bond vibration. That means these are unsaturated lipids. That is, already easy to understand. And then we just constructed a library of all sorts of unsaturated lipids, starting from 16.1 to 22.6, which has 6 double bonds. So as we increase the number of double bonds here, we can see that there is a linear correlation, and our data fits at, with the di-unsaturation. I think I'm almost about time, so I'm going to finish in 1 minute. So anyways, what we can do is that we have already identified that the lipid 1 is actually a linoleic acid with 2 double bonds. And we also, from the literature, it is very clear that linoleic acid is actually the precursor for prostaglandins. So this means that in the case of normal cells, there is, in the case of cancer cells, there is less abundance of this lipid because it's actually leading to the formation of more and more prostaglandins. And however, all this while what we have discussed is that Raman spectroscopy can discriminate very well, but again, there are lots of human interference in terms of data analysis. So how can we actually avoid that? We try to address that by using neural networks, where this is the framework. I don't want to get into details of how, because probably it will take much longer. Anyways, we do some kind of data pre-processing and increase large amount of data by augmentation, and then we build the model and then we try to evaluate. So anyways, what we can actually do, we obtain is that we would obtain accurate classification with no or minimal human interference with the same data set with about 98% accuracy. So we understand the molecular information. We also understand that it is possible to use Raman spectroscopy for very rapid analysis of single cells with no human interference. Thank you so much for your patience.
Thank you so much for this insightful talk on how machine learning coupled with Raman spectroscopy can be applied for diagnostics and healthcare. And this ends towards the end of our sessions. So all the participants please join room A. Yes, for all the speakers in this session, even taking your interest and participation, and two announcement that participation certificate will be sent to all of you by email. And if anyone is looking for hard copy, please